Before animals learned to fight, before they learned to hide, before they learned to see, there was something else, something older, something stranger. A world so alien it barely registers in our imagination, yet so fundamental that without it, nothing we know would exist. This is the story of the Ediacaran period, the deep, silent chapter that came before everything. The chapter that most people have never heard of, and the one that changed life on Earth forever. Please let us know in the comments where you are watching this history video and what time you are watching it. And be sure to subscribe to the channel for future videos. Close your eyes for a moment and try to imagine an ocean with no fish, no whales, no coral reefs, no sharks cutting silently through the dark water, no crabs scuttling across the sand, no jellyfish pulsing through the current, no sound of movement, no chase, no predator, no prey, just water, just chemistry. Just the slow, ancient rhythm of tides pushed by a moon that hung closer to the Earth than it does today, pulling the oceans with a heavier hand, stirring the world with a kind of gravitational patience that we can barely comprehend from where we stand now. Go back far enough and the ocean is not a place of drama. It is a place of stillness, a place of waiting. But here is the thing about waiting, it is never truly empty. Even in the deepest stillness, something is happening. Even before the first eye opened on this planet, before the first creature lunged toward another in hunger or desperation, life was already doing something remarkable. Life was experimenting, life was reaching, life was becoming. That is what this documentary is about. We are going back more than 540 million years. We are going back to time before the Cambrian period, before the so-called explosion of animal life that most people learn about when they first study the history of biology. We are going back to the period that came before that explosion, the period that made that explosion possible, a time in Earth's history known as the Ediacaran. The Ediacaran period lasted roughly from about 635 million years ago to approximately 538 million years ago. That is nearly 100 million years of Earth's history. 100 million years during which life on this planet was doing something it had never done before and would never quite do again, assembling itself into complex multicellular forms for the very first time, building bodies out of cooperating cells, experimenting with shape and structure and behavior in ways that were genuinely, profoundly new. And yet most people have never heard of it. Most people, when they think about ancient life, jump straight to the dinosaurs. Some go a little further back and think about the great marine creatures of the Mesozoic seas, the mosasaurs, the plesiosaurs, the ammonites spiraling through warm, shallow water. A few go further still and think about the Cambrian, about trilobites in the Burgess Shale, about that famous explosion of animal forms that Darwin himself found troubling, because it seemed to happen so suddenly in the fossil record. But before all of that, before the Cambrian, before the first trilobite, before the first creature with a recognizable head and tail, there was the Ediacaran. And the Ediacaran is not just a footnote, it is not just a prelude. It is one of the most profound and mysterious chapters in the entire 4 billion year story of life on Earth. We are going to spend the next 2 hours inside that chapter. We are going to walk the ancient seafloors. We are going to meet the organisms that lived there, creatures so strange that scientists have argued for decades about whether they were animals, plants, fungi, or something else entirely, something with no equivalent anywhere in the living world today. We are going to watch them live and feed and move and die. We are going to follow the slow, systemic transformation that eventually ended their world and gave rise to ours. And we are going to sit with the deeper philosophical question that the Ediacaran always seems to raise. What does it mean for something to come before everything we know? Because here is the truth. The Ediacaran world did not fail. It did not simply lose a competition it was ill-equipped to win. It was a complete world, a functioning world. A world with its own internal logic, its own ecological relationships, its own beauty. And understanding that world, really understanding it, not just cataloging its fossils, but inhabiting it imaginatively and intellectually, is one of the great scientific and philosophical adventures available to us. So let us begin at the beginning. Or rather, let us begin just before the Ediacaran, because to understand what the Ediacaran was, we first need to understand what came before it. We need to understand the world that had to be transformed before complex multicellular life could take its first real steps. The Earth is approximately 4.5 billion years old. That number is so large it has become almost meaningless to us as human beings. We hear it, we nod, we move on. But try for a moment to sit inside that number. Try to feel the weight of it. If you compressed all of Earth's history into a single 24-hour day, modern humans would not appear until the last 2 seconds before midnight. The dinosaurs would not show up until about 11:30 in the evening. The Cambrian explosion of animal life, that famous moment when recognizable animal forms first proliferated across the fossil record, would happen around 9:30 at night. And the Ediacaran period, the subject of this entire documentary, would begin around 9 in the evening. Everything before 9 in the evening on this imaginary day, more than 18 hours of 24, would be the story of microscopic life. Single cells, prokaryotes without nuclei, then, eventually, eukaryotes with nuclei, then colonies of cells beginning to work together, nothing you could see with the naked eye. Nothing with a front and a back, nothing with specialized tissues, nothing with the capacity to move through the world as a coherent organism pursuing goals, however simple. For most of Earth's history, life was invisible. Life was chemical. Life was a vast, patient, planetary-scale process of microscopic transformation, reshaping the oceans and the atmosphere over billions of years, laying down the chemical and biological infrastructure that everything else would eventually depend on. And in some ways, that invisible life was the most consequential life that ever existed on this planet, because here is what those billions of years of microbial life actually accomplished. In the beginning, Earth's atmosphere had virtually no free oxygen. The early ocean was a reducing environment, chemically very different from the oxygenated world we know today. Life, in its earliest forms, did not need oxygen. In fact, for the first forms of life on Earth, oxygen was a poison, a toxic byproduct of a metabolic process that would eventually reshape the entire planet. That metabolic process was photosynthesis. Around 3 billion years ago, certain bacteria, the cyanobacteria, developed the ability to harvest energy from sunlight and use it to split water molecules, releasing oxygen as a byproduct. And for hundreds of millions of years, that oxygen was immediately absorbed by iron dissolved in the ancient ocean, forming the great banded iron formations we see in rocks around the world today, layers of rust, essentially, laid down over vast stretches of time as the cyanobacteria slowly, incrementally oxygenated the water around them. But eventually, around 2.5 billion years ago, the iron in the ocean began to run out. The oxygen had nowhere to go. It began to build up in the water, and then it began to escape into the atmosphere. This event, the Great Oxidation Event, was arguably the greatest ecological catastrophe in the history of life on Earth. For the anaerobic organisms that had dominated the planet until then, the rising oxygen levels were lethal, mass extinction on a planetary scale. The microbes that had shaped Earth's chemistry for over 1 billion years were largely destroyed by the byproduct of their own planetary neighbors. But out of that catastrophe came opportunity. The new oxygen-rich environment opened up entirely new metabolic pathways. Organisms that could use oxygen, that could breathe it, essentially, in a biochemical sense, could extract far more energy from the same amount of food than their anaerobic predecessors. This metabolic windfall would eventually power everything from fish to mammals to us, but that was still billions of years away. For hundreds of millions of years after the Great Oxidation Event, life remained fundamentally microbial. There were complex cells, eukaryotes, cells with nuclei, cells with internal compartments appearing around 2 billion years ago. There were early multicellular forms, but nothing that looks like what we would recognize as an animal, nothing with a body plan. Nothing with tissues, nothing with coordinated behavior. The world was covered in microbial mats. This is an image that will become very important as we move through this documentary, so let us spend a moment with it. Microbial mats were, and in some isolated environments still are, thick, layered communities of microorganisms, primarily cyanobacteria and other photosynthetic bacteria that grew across the seafloor and the shallow margins of ancient oceans. They were not dramatic. They were not visually impressive in the way we might imagine life to be. They were more like living carpets, thin, coherent layers of microbial community, sometimes just millimeters thick, sometimes building up into more substantial structures like stromatolites, which are layered, dome-shaped or columnar formations that still exist in a few places today, and that represent some of the oldest evidence of life on Earth. These microbial mats were the foundation of the Ediacaran world. They were the substrate on which the first complex organisms would eventually make their home. They were the food source, the structural anchor, the chemical environment, the everything for the strange creatures that were about to appear. But first, the world had to go through something extraordinary, something violent and strange and almost unimaginable in its totality. The world had to freeze. Before the Ediacaran, in the period geologists call the Cryogenian, roughly from around 720 million to 635 million years ago, Earth experienced the most severe glaciations in its history. These events are collectively known as Snowball Earth, and the name is not an exaggeration. There is strong geological evidence that during the Cryogenian, glaciers extended all the way to the equator. The entire planet, or very nearly the entire planet, was encased in ice. The oceans froze over. The continents were buried under kilometers of glacial ice. The photosynthetic productivity of the planet essentially collapsed because sunlight could not penetrate the thick ice covering the ocean's surface. How life survived these events is still not entirely clear. It almost certainly did not survive everywhere. Many lineages were almost certainly lost. But life did survive, perhaps in isolated pockets of volcanic warmth, around hydrothermal vents, or in thin films of liquid water at the base of the ice where pressure prevented complete freezing. Life persisted, as it has a way of doing, in the cracks and margins of even the most hostile conditions. And when the Snowball Earth events ended, when volcanic activity, continuing relentlessly beneath the ice, gradually raised carbon dioxide levels in the atmosphere enough to trap heat and begin melting the ice, the world that emerged was chemically and physically transformed. The melting of all that glacial ice released vast quantities of nutrients into the ocean. The conditions for biological innovation were, in some fundamental sense, reset. This is the context in which the Ediacaran begins. 635 million years ago, emerging from the deep freeze of the cryogenian into a transformed ocean, into a world still sorting itself out chemically, comes the Ediacaran Period, and with it eventually the first complex multicellular organisms with bodies. The transition was not instant. Even after the glaciations ended, it took tens of millions of years for complex life to emerge and diversify. The first part of the Ediacaran was still a microbial world, still dominated by the same mat-forming communities that had characterized the planet for billions of years. But something was changing. Something in the chemistry of the ocean, in the oxygen levels, in the nutrients available, in the evolutionary pressures, something was beginning to make new things possible. And around 575 million years ago, the fossil record begins to show us something new. Something that makes paleontologists stop and stare, something that once you see it, you cannot unsee. The first Ediacaran organisms. Let us talk about how we know any of this, because one of the most remarkable things about the Ediacaran period is not just the organisms themselves, but the nature of the evidence we have for them. The story of how we discovered the Ediacaran world is itself a remarkable scientific journey, full of confusion and revision, and the kind of intellectual humility that the best science always requires. The first Ediacaran fossils were found long before anyone understood what they were. In the 1930s and 40s, geologists working in South Australia found strange impressions in rocks from the Flinders Ranges, disc-like forms and elongated ribbed shapes that were clearly biological in origin, but that didn't match anything in the known fossil record. For years, there was no clear framework for understanding them. Some researchers thought they might be trace fossils, the tracks or burrows of organisms rather than the organisms themselves. Some thought they might be algae. Some thought they might be some kind of colonial organism like a coral, though far too old for any known coral. It was not until the 1950s and 60s that these Australian finds were properly connected to similar fossils being found in other parts of the world, in England, in Russia, in Namibia, and the concept of a distinctive Precambrian multicellular biota began to take shape. The period itself was eventually named after the Ediacaran Hills in South Australia, where some of the most important early specimens were found. And then in 1967, a discovery was made in Newfoundland that would become one of the most significant fossil sites in the world. The cliffs of Mistaken Point, on the southeastern tip of the island of Newfoundland, expose ancient seafloor surfaces that were buried by volcanic ash approximately 565 million years ago. And on those surfaces, preserved in extraordinary detail by the rapid burial that the ash provided, are the impressions of entire communities of Ediacaran organisms, hundreds of individuals, frozen in their ecological relationships exactly where they lived and died. Mistaken Point is remarkable for many reasons. The fossils there are among the oldest known Ediacaran body fossils. The preservation is exceptional, not just individual specimens but whole communities preserved in situ, giving scientists a genuine ecological snapshot rather than isolated fragments. And the organisms preserved there are among the strangest and most alien in the entire fossil record. They are not animals, they are not plants, they are not fungi. They are not any category that fits neatly into the classification systems we use for modern life. They have been described variously as quilted mattresses, as fractal fronds, as creatures with no modern equivalent in any kingdom of life. And the more carefully scientists have studied them, the more they have come to suspect that some of these organisms may represent an entirely distinct experiment in multicellular life, one that left no descendants, one that tried a fundamentally different approach to being large and complex, and one that eventually failed in a competitive sense, though failed is perhaps not quite the right word. Let us meet some of these organisms. Let us walk the ancient seafloor of the Ediacaran and try to understand who was there. Imagine you are on the seafloor, not a modern seafloor, an Ediacaran seafloor, somewhere around 565 million years ago. The water above you is cold and relatively shallow. Light filters down from above, though not in great abundance. You are in what would be described as the twilight zone of the ancient ocean, not in the deepest dark, but not in the bright upper waters either. The seafloor beneath you is covered in a thick, continuous mat of microbial life. It is not dramatic to look at. It is dark, slightly greenish or brownish, textured like very fine velvet or felt. It has a faint smell of sulfur and decay. It is alive in the most fundamental sense, billions of individual cells, working together in complex chemical cycles, cycling nutrients through the sediment and the water above. And growing from that mat or resting on it or pressed against it are the Ediacaran organisms. The first thing you notice is that there are no eyes looking at you. Nothing moves suddenly when you arrive. There is no instinctive flight response, no predator-prey tension in the air. The organisms simply exist, growing, absorbing, potentially sensing in ways we cannot fully understand, but doing so in a register completely different from anything in the modern ocean. Rising from the mat in front of you is what looks, at first glance, like a fern frond. It is long and flat, slightly curved, with a branching structure that repeats at smaller and smaller scales, a fractal pattern, repeating itself from the level of the whole organism down to the smallest visible branch. It is attached to the seafloor at one end and it sways slightly in the weak current. This is Charnia. Charnia is one of the oldest known Ediacaran organisms. First described from fossils found in England's Charnwood Forest in the 1950s, found, remarkably, by schoolchildren who recognized that the impressions in the rock were unusual. Charnia has since been found at Mistaken Point and in other Ediacaran deposits around the world. It is among the oldest complex organisms known from the fossil record, living around 565 to 575 million years ago. Charnia belongs to a group of Ediacaran organisms known as rangiomorphs, and the rangiomorphs are perhaps the most alien life forms we know of from any period of Earth's history. Their body plans are built on a principle of self-similar branching, the same basic structure repeated at multiple scales, the way a fern frond is made of smaller frond-like units, or the way a coastline looks similar at every scale of magnification. This is the mathematical property known as fractality, and it appears to have been a fundamental organizing principle for rangiomorph anatomy. What were the rangiomorphs doing? How did they feed? This question has been the subject of considerable scientific debate. They had no mouths. They had no digestive systems in any sense that we could recognize. They had no moving parts that we can identify. And yet they were large. Some rangiomorphs were more than a meter in length, which by the standards of the Ediacaran was genuinely enormous, and they maintained their complex organized structures, which requires energy. The leading hypothesis is that rangiomorphs fed by absorbing dissolved organic carbon directly from the water around them. Essentially, they fed by osmosis through their body surfaces. Their fractal branching structure would have been perfectly adapted for this purpose, because the fractal design maximizes surface area while minimizing the volume of body tissue that needs to be maintained. A rangiomorph with its endlessly repeated branches had an enormous surface area relative to its size, far more surface area than any smooth-bodied organism of comparable dimensions, and all of that surface area could be used for absorbing nutrients from the water. This is a feeding strategy that has no real equivalent in the modern animal world, though it has faint echoes in the way some modern organisms absorb nutrients. It is a strategy that works in an environment where organic carbon is abundant in the water, and in the Ediacaran Ocean, with its vast microbial mats releasing organic molecules into the water column. Such a strategy may have been genuinely effective, but Charnia is just one of the rangiomorphs at Mistaken Point. Look around and you see others. Bradgacia with its rounded, bushy form like a tight cluster of branches spreading in multiple directions. Fractifusus, elongated and spindle-shaped, lying on the seafloor rather than standing upright, its fractal branches spreading laterally. Hemilora with a central disc and radiating structures extending outward like the spokes of a wheel. The diversity of forms at Mistaken Point is striking. These are not all the same organism. They are different species, different body plans, different orientations, some upright, some prostrate, some perhaps changing orientation depending on current conditions. And they live together in communities, in specific ecological relationships with one another and with the seafloor beneath them. When paleontologists have analyzed the spatial distribution of organisms at Mistaken Point, they have found that different species tend to cluster in different ways, and that the overall community structure is not random. Some species are found preferentially near others. Some species seem to occupy particular ecological niches on the ancient seafloor. This is ecology, real, functioning ecology, not just a random assemblage of organisms, but a structured community with its own internal organization. This might seem obvious, but it is actually a profound and important observation. The existence of ecological structure in the Ediacaran, the fact that these organisms were not just randomly distributed but organized in meaningful ways, tells us that even in this earliest chapter of complex multicellular life, the processes of ecological organization that we take for granted in modern ecosystems were already operating. Life was already structuring itself, already competing perhaps for space and resources. Already in the complex dance of interaction that defines any functioning ecosystem. Now let us step forward in time, not by much, perhaps 10 to 15 million years, and let us move to a different part of the ancient ocean. We are now somewhere around 555 to 560 million years ago, and we are looking at a different kind of fossil site, different kinds of organisms. The Ediacaran fossil record comes from many parts of the world: South Australia, Newfoundland, the Russian White Sea coast, the Nama Group in Namibia, the Lanshan Formation in China, among others. Each of these sites preserves a somewhat different community of organisms, from somewhat different time periods, under somewhat different environmental conditions. Together they allow us to build up a picture of the Ediacaran world through time, not perfectly, not without gaps and ambiguities, but with increasing resolution as more fossils are found and more careful analysis is done. The White Sea coast of Russia has yielded some of the most important and diverse Ediacaran fossils. The organisms preserved there lived somewhat later than those at Mistaken Point. They are from the final 50 or 60 million years of the Ediacaran period, and they show us a world that has changed significantly from the rangiomorph-dominated seafloors of earlier times. Here, we begin to see something new, something that, if you squint at it in the right way, begins to look familiar. We begin to see the emergence of body plans that have bilateral symmetry, that have a definable front and back, a left side and a right side, a head end and a tail end. And with bilateral symmetry comes the possibility of directional movement. With directional movement comes the possibility of behavior. With behavior comes the possibility of ecology as we recognize it. This is where one of the most fascinating and debated organisms in the entire Ediacaran fossil record appears. Dickinsonia. If you were to see a Dickinsonia fossil for the first time without knowing what it was, you might initially mistake it for a strange kind of leaf or perhaps an oddly segmented structure left by some physical process, a ripple pattern in the sediment perhaps. Dickinsonia is typically oval or ribbon-shaped, ranging in size from a few millimeters to nearly 1.5 meters in length, though most specimens fall somewhere in the middle of that range. Its surface is divided into a series of bands or modules, not quite segments in the way an earthworm or a trilobite has segments, but a series of repeating units arranged in a bilaterally symmetrical pattern, meeting at a central axis that runs the length of the body. At first glance, Dickinsonia seems to have a definite front end and a back end, though distinguishing which has been a matter of considerable scientific debate. It appears to have no obvious sensory organs, no eyes, no apparent antennae, no structures that obviously served for detecting the environment. It had no obvious mouth or digestive system in the conventional sense. For decades after its discovery, Dickinsonia was one of the most argued over organisms in paleontology. Scientists proposed that it was a cnidarian related to modern jellyfish and sea anemones. They proposed it was an annelid worm. They proposed it was a fungus. They proposed it was a lichen. Some researchers placed it in the so-called Vendobionta, a proposed kingdom of extinct organisms with no modern relatives, a group defined specifically to hold the Ediacaran organisms that fit nowhere else. The question of what Dickinsonia actually was, biologically and evolutionarily, remained genuinely open for most of the 20th century. It is a perfect illustration of why the Ediacaran is so intellectually challenging. These organisms are so different from anything alive today that our normal categories simply do not fit them comfortably. We keep reaching for a modern analog and keep finding that no modern analog quite works. But then came a remarkable scientific breakthrough in 2018, published in the journal Science. A team of researchers, led by Jochen Brox of the Australian National University, managed to extract organic molecules, specifically biomarkers called sterols, from exceptionally preserved Dickinsonia specimens found in Russia. The Dickinsonia specimens were so well preserved that organic material from the organism itself had survived, despite being over 550 million years old. And the molecules they found were cholesterol-like sterols, specifically the kind of sterols characteristic of animals. Dickinsonia, the analysis suggested, was an animal, or at least it was on the animal side of the great divide between animals and other kinds of eukaryotes. It was making the same biochemical machinery as animals make. Its cells were fundamentally animal-like in their chemistry. This was a significant result. It placed Dickinsonia firmly within the animal lineage, not as a relative of any particular modern animal group, perhaps, but as genuinely animal in the most fundamental biochemical sense. And it moved the origin of animals further back in time than many researchers had previously accepted, suggesting that the animal lineage was already present and already diversified in the Ediacaran world. But Dickinsonia's status as an animal does not resolve all the mysteries about it. If anything, it deepens them, because if Dickinsonia was an animal, it was a deeply strange animal. It had no hard parts, it had no clear head with centralized sensory equipment, it had no obvious limbs or appendages. It lived on or very near the seafloor, and evidence suggests that it moved slowly, gradually, repositioning itself across the microbial mat, and that it fed on the mat material as it moved. The evidence for feeding behavior in Dickinsonia is one of the most elegant and telling findings in Ediacaran paleontology. Some specimens have been found in association with patches of disturbed microbial mat areas where the mat texture is different from the surrounding material, consistent with the organism having absorbed or consumed the mat material from beneath its body, much the way a very primitive form of grazing might work. And when the same individual is found in different positions at different moments of a fossil sequence, when we can reconstruct movement from the fossil record, the organism appears to have been moving toward fresh, undisturbed mat and away from areas it had already processed. This is behavior. This is ecological behavior, not sophisticated behavior, not behavior that involves a nervous system in any sense we would recognize, but behavior nonetheless. Dickinsonia was moving through its world in a purposeful way, exploiting resources, navigating its environment. And this represents something genuinely new in the history of life. Before Dickinsonia and organisms like it, the ecological interactions of the living world had been fundamentally chemical, a game of nutrients and molecules, of absorption and excretion, of chemical gradients and chemical responses. With organisms like Dickinsonia, we are beginning to see the emergence of something different, organisms that move through their environment in directed ways that make choices, however rudimentary, about where to go and how to position themselves. The seeds of behavior. The Ediacaran world was not static. Over the roughly 100 million years of the period, it changed. It changed biologically as organisms evolved and new body plans appeared. It changed ecologically as the relationships between organisms and their environment became more complex. And it changed physically as the oceans and the atmosphere continued to evolve in their chemistry and in their patterns of circulation. One of the clearest trends across the Ediacaran is the gradual increase in biological diversity. The earliest Ediacaran communities, like those at Mistaken Point, are dominated by the rangeomorphs, those strange fractal frond-like organisms. But as we move toward the later part of the period, we see an increasing variety of forms. We see organisms with more definite bilateral symmetry. We see the first hints of what will eventually become recognizable animal body plans. We see ecological relationships becoming more complex, and we see the beginning of something that will fundamentally transform the world. We see the beginning of movement. Not just the slow, sessile existence of organisms attached to the seafloor, swaying in the current. Not just the gradual repositioning of organisms like Dickinsonia across the mat surface. We begin to see in the trace fossil record, the record of tracks, trails, and burrows left by organisms moving through or across the sediment, evidence of more active, more directed movement. We begin to see the first animals making their mark on the physical structure of the seafloor. This is enormous. This is not just a biological development, it is an ecological revolution. When organisms begin to move through the sediment, burrowing into it, mixing it, processing it, they transform the chemistry and the structure of the seafloor in ways that have cascading consequences for everything else that lives there. But we are getting slightly ahead of ourselves. Before we can fully appreciate the ending of the Ediacaran world, we need to spend more time inside it. We need to meet more of its inhabitants. We need to understand what was actually happening in these ancient oceans on a day-to-day, organism by organism basis. So let us go back. Let us meet more of the Ediacaran cast. Alongside the rangeomorphs and Dickinsonia and the White Sea assemblage, we find a remarkable organism called Kimberella. and Kimberella is in many ways the most important organism in the entire Ediacaran fossil record for understanding the transition from the Ediacaran world to what came after. Kimberella is not large by modern standards. Most specimens are a few centimeters to perhaps 10 or 15 centimeters in length. Its body appears to be roughly oval in shape, with a somewhat rigid upper surface and a softer, more flexible underside. It has what appears to be a clearly defined front end and back end. And associated with many Kimberella fossils in the sediment around and beneath them are distinctive scratching marks, traces that look for all the world like the scratching action of a rasping feeding organ. In modern mollusks, snails, and their relatives, the rasping feeding organ is called the radula. It is a structure covered in small tooth-like projections that the animal uses to scrape algae or biofilm off hard surfaces, drawing the material into the mouth. The traces associated with Kimberella specimens look strikingly similar to the kind of traces a radula would make. Fan-shaped patterns of scratches, overlapping arcs suggesting a repeated rasping action directed toward fresh microbial mat. If this interpretation is correct, and it is not universally accepted, though it is widely supported, then Kimberella may be one of the oldest known relatives of the mollusks. It may have been an early member of the lineage that eventually gave rise to snails, clams, squid, and octopuses. It would push the origin of that lineage back into the Ediacaran, establishing that the major animal body plans were not just beginning to emerge at the very end of the Ediacaran, but were already diversifying well within it. But what is most significant about Kimberella for our purposes is not its possible taxonomic placement. It is what Kimberella tells us about the ecological transformation of the Ediacaran world in its later stages. Kimberella was not absorbing nutrients passively through its body surface, the way the rangiomorphs appear to have done. Kimberella was actively grazing. It was moving across the microbial mat, scraping material off the surface, ingesting it, processing it internally. It was a consumer in a much more recognizable sense, an organism that moved to its food and actively harvested it. This represents a fundamental shift in the ecological dynamics of the Ediacaran world. When passive absorbers give way to active grazers, the relationship between organisms and their environment changes qualitatively. A passive absorber takes what is available in the water around it. An active grazer chooses where to go. An active grazer has preferences, has behaviors, has something, however rudimentary, that we might call agency. And active grazers create selection pressure. They put pressure on the microbial mat to evolve defenses. They create a world in which being palatable is a disadvantage. They begin to set in motion the arms race between consumers and resources that ultimately defines so much of modern ecology. The Ediacaran is, in many ways, the world before that arms race had really gotten going. But Kimberella and organisms like it are the first sign that the arms race was beginning. They are the first heralds of the world to come. Not all Ediacaran organisms were soft and formless. Some had what we might describe as semi-rigid structures, not true mineralized shells or bones, but organized body walls that maintained their shape against the forces of water and current. And in the later parts of the Ediacaran, we begin to see the first experiments with biomineralization, with using calcium carbonate or other minerals to build hard, protective structures. The Nama group in Namibia is one of the most important sites for understanding the final stages of the Ediacaran period. The organisms preserved there lived in the last 20 or 30 million years of the Ediacaran, and they show us a world that is in many ways already beginning to look different from the earlier Ediacaran communities. Among the Namibian Ediacaran organisms is a creature called Cloudina. Cloudina is remarkable for a specific reason. It had a hard, tube-like shell. It was, as far as we know, one of the earliest animals to build a mineralized skeleton, to use calcium carbonate to construct a protective outer covering. The Cloudina shell was not particularly thick or robust. It was more like a series of nested cones, each slightly larger than the last, forming a tube that the animal lived inside. But it was hard. It was mineral. It was a physical barrier between the organism's soft body and the outside world, and it appears to have been necessary. Because some Cloudina specimens show evidence of predation, tiny holes bored through the shell wall, consistent with some unknown predator drilling through the shell to reach the soft tissue inside. These are the oldest known examples of predatory drilling in the fossil record. They tell us that by the end of the Ediacaran, predation was not just beginning, it was already creating selection pressure for defensive structures. The arms race was underway. The emergence of predation, real targeted predation, with predators actively hunting and killing other organisms, is one of the most significant ecological transitions in the history of life. Before predation became a significant ecological force, life had no need to invest energy in hard protective structures, in fast locomotion, in sensory systems for detecting danger, in behavioral strategies for escape and evasion. After predation became significant, everything changed. The economic logic of building a body shifted dramatically when the primary threat to that body's continuation was no longer starvation or environmental stress, but another animal with the intention of eating you. Cloudina and its contemporaries at the end of the Ediacaran were living in a world where that transition was happening. They were on the front line of it. And the response they show us, the investment in hard mineralized structures, the first shells, is a preview of the enormous radiation of skeletal animals that would occur in the Cambrian, just a few tens of millions of years later. There is something deeply poignant about Cloudina when you think about it in this way. Here is an organism, living at the edge of the world it has always known, beginning to respond to a new kind of threat that world had never had to contend with. Ediacaran world, for most of its existence, was a world without significant predation. It was a world of slow ecological processes, of passive absorption and gradual grazing, of the measured rhythms of growth and reproduction and death without violence. And into that world, predation was arriving inexorably, driven by the same evolutionary processes that had produced all the other innovations of the Ediacaran, but producing something qualitatively different from what had come before. The shell of Cloudina is a response to fear, or rather, since Cloudina almost certainly had no conscious experience of fear in any sense we would recognize, the shell is a response to selection pressure created by predation. It is evolution's answer to the question, what do you do when something is trying to eat you? You build armor. And that armor, that first, relatively thin, somewhat fragile, but genuinely mineral shell, is the ancestor of every shell, every exoskeleton, every bone, every tooth that has ever existed on this planet. The principle encoded in Cloudina's tiny nested cones of calcium carbonate is the same principle at work in a trilobite's exoskeleton, a crab's carapace, a dinosaur's bone, and the human skull. The principle that biological material can be made hard, can be made structural, can be made to serve as protection and support. In this sense, Cloudina is one of the most consequential organisms that ever lived, not because it was spectacularly successful. It existed for perhaps 20 or 30 million years and then disappeared, like most Ediacaran organisms. But because the evolutionary experiment it represented, the experiment of biomineralization, was the experiment that worked. It was the experiment that the Cambrian would take and run with, proliferating into the enormous diversity of shelled and skeletal animals that have dominated the fossil record ever since. Let us step back from the individual organisms for a moment and think about the Ediacaran world as a whole system. Because one of the most important insights that modern paleontology has brought to our understanding of the Ediacaran is precisely this systems-level view. The recognition that the organisms of the Ediacaran were not just individual entities, but parts of functioning ecosystems, and that those ecosystems had their own internal dynamics, their own feedbacks, their own vulnerabilities. The Ediacaran ecosystem, particularly in its earlier phases, was built around the microbial mat. The mat was, in ecological terms, the primary producer, the foundation of the food web, the structural substrate of the seafloor community. Everything else depended on it in one way or another. For the rangiomorphs, the mat provided the physical base from which they grew upward, and the organic molecules they absorbed from the water above were in part produced by the decomposition of mat material. For organisms like Dickinsonia, the mat was directly the food source. They absorbed it from beneath their bodies as they moved across it. For later organisms like Kimberella, the mat was the material being scraped and consumed by the radula-like feeding organ, and the mat itself was not passive. It was a living community, constantly growing, constantly producing organic material, constantly cycling nutrients through the sediment and the water. It was the engine of the Ediacaran ecosystem, and as long as it functioned, it sustained the community of complex organisms living upon and within it. But the mat's dominance was contingent on specific conditions. The mat required a relatively stable seafloor, one that was not constantly being disrupted and mixed. It required specific chemical conditions in the water and sediment. It required, in particular, the absence of the kind of bioturbation, the physical mixing of sediment by burrowing organisms, that we take for granted in modern seafloors. Modern seafloors are profoundly bioturbated. Almost everywhere in the modern ocean, the top several centimeters of sediment are mixed and reworked by organisms, worms, clams, sea cucumbers, and hundreds of other species that burrow into the sediment, process it, turn it over, change its structure and chemistry. This constant biological mixing is so pervasive in modern oceans that it completely prevents the formation of the kind of stable, layered microbial mats that characterize the Ediacaran seafloor. In the Ediacaran, bioturbation was minimal. The organisms doing any burrowing at all were simple, small, and relatively ineffective at deep mixing. The seafloor was, by modern standards, almost undisturbed. And this allowed the microbial mat to do what it does, to grow in continuous layered sheets across the seafloor, to maintain its chemical stability, to persist as the defining feature of the benthic environment. The stability of the Ediacaran ecosystem, in other words, was directly dependent on the absence of significant bioturbation. And as the Ediacaran progressed, as organisms evolved that were capable of more effective burrowing, that stability was gradually eroded. The mat was disrupted. The chemical environment of the seafloor changed, and the organisms that had depended on the mat, that had built their entire ecological existence around the mat's stability and productivity, found themselves in an environment that was no longer suited to them. But this is the ending. This is the gradual fading out of the Ediacaran world, and we are not quite ready to discuss that yet. There is still much of the world to explore, still organisms to meet, still ecological dynamics to understand. Let us talk about the Chinese Ediacaran fossils, because the fossil record of the Ediacaran is not confined to Australia and Russia and Namibia and Canada. China has also yielded remarkable Ediacaran material, and some of the most extraordinary finds come from sites in Yunnan and Anhui provinces. The Lanshan biota from the Lanshan Formation in Anhui province is dated to approximately 600 million years ago, making it one of the oldest known Ediacaran macrofossil assemblages. The organisms preserved there include a variety of frond-like and leaf-like forms, as well as some that are harder to categorize. What is particularly striking about the Lanshan biota is the evidence it provides for ecological differentiation, for organisms occupying different positions in the water column and on the seafloor, suggesting that even at this early stage, the Ediacaran community was more ecologically complex than a simple assemblage of identical organisms passively absorbing nutrients. Even more remarkable is the Daoshenchu Formation, also in China, which preserves microscopic fossils, some of the smallest and most exquisitely detailed fossils anywhere in the Ediacaran record. The Daoshenchu microfossils include what appear to be animal embryos, clusters of cells in various stages of division that resemble the early embryonic development of animals. If these are indeed animal embryos, they push the origin of the animal lineage back to somewhere around 600 to 630 million years ago, deep in the Ediacaran, close to its beginning. The interpretation of the Daoshenchu microfossils has been controversial, as is so much in Ediacaran science. Some researchers argue that they are indeed animal embryos. Others have proposed that they are the reproductive stages of giant unicellular organisms, or algae, or some other kind of organism not previously recognized in the fossil record. The debate continues, as it should. The evidence is genuinely ambiguous, and the stakes of interpretation are high. But the existence of these tiny, perfectly preserved cellular structures in the Daoshenchu Formation points to something important about the nature of Ediacaran science. The story of early complex life is not only told in the large, visible impressions of organisms like Dickinsonia and Charnia. It is also told at the cellular level, in the chemistry of ancient rocks, in the molecular remnants of organisms that lived and died hundreds of millions of years ago. Understanding the Ediacaran requires working at multiple scales simultaneously, from the broad ecological patterns of whole fossil communities to the molecular details of individual biochemical pathways. This is what makes Ediacaran science so intellectually demanding and so endlessly fascinating. It is not just a matter of identifying new species and describing them. It is a matter of developing new analytical tools, new conceptual frameworks, new ways of reading ancient information encoded in rock and chemistry. Every decade brings new techniques, and with them new insights, new revisions, new appreciations of the complexity of what was happening more than 500 million years ago. We have been speaking mostly about what we can see, the macroscopic organisms, the body fossils, the trace fossils. But there is another dimension of the Ediacaran world that is just as important and far harder to access: the chemistry of the ancient ocean. The Ediacaran ocean was chemically different from the modern ocean in important ways. Most significantly, it was less oxygenated. The Great Oxidation Event had put significant oxygen into the atmosphere over 2 billion years ago, but the oxygenation of the deep ocean was a much slower process. Throughout much of the Ediacaran, the deep ocean remained poorly oxygenated or even anoxic, without significant dissolved oxygen. Only the upper layers of the ocean and the shallow coastal environments had the oxygen levels needed to support the metabolically active organisms we have been discussing. This matters enormously for understanding the distribution and ecology of Ediacaran organisms. The communities we have been examining, the rangiomorphs at Mistaken Point, the diverse assemblages of the White Sea, the organisms of the Namibian Nama group, all lived in shallow water environments or at intermediate depths where oxygen was available. The deep ocean of the Ediacaran was largely still a microbial world, still chemically reducing, still dominated by the same biochemical processes that had characterized it for billions of years. The shallow, oxygenated zones of the Ediacaran Ocean were, in this sense, a new ecological frontier. They were environments where the oxygen levels were high enough to support the energy-intensive business of building and maintaining a complex multicellular body. And the Ediacaran organisms colonized those environments with remarkable creativity, evolving a diversity of forms and strategies in the relatively small area of ocean that was energetically available to complex life. As the Ediacaran progressed, oxygen levels in the ocean appear to have risen in complex, non-linear ways. There were probably pulses of oxygenation, periods when oxygen levels rose significantly, followed by periods when they fell back. These oscillations may have driven some of the evolutionary dynamics of the Ediacaran, opening up new ecological space during oxygenation events and then placing pressure on newly established communities during subsequent falls in oxygen. The pattern of Ediacaran evolution, in other words, may have been partly a function of the evolving chemistry of the ancient ocean, not just of the inherent evolutionary capabilities of the organisms themselves. This interplay between biological evolution and environmental change, between life and the physical world that contains it, is one of the deepest themes in the history of life. Life does not evolve in a vacuum. It evolves in response to the environment, and the environment is itself partly a product of life's activity. The cyanobacteria that oxygenated the ancient ocean were doing something that made the world profoundly different, and in doing so, they were laying the groundwork for the evolution of everything that followed. The Ediacaran organisms, in their turn, were doing things that made the world different, changing the chemistry of the seafloor sediment, contributing to the organic carbon cycle, beginning the process of bioturbation that would eventually transform the seafloor into the biologically mixed environment of the modern ocean. Life shapes the world, and the world shapes life. This is the great reciprocal story that runs through all of Earth's history, and it runs through the Ediacaran period with particular clarity. Let us turn now to one of the most beautiful and evocative aspects of Ediacaran science, the fossil sites themselves. Because the places where Ediacaran fossils are found are not just scientifically important, they are in their physical reality places of extraordinary power and strangeness. Mistaken Point, on the southeastern tip of Newfoundland, is now a World Heritage Site, a recognition of its extraordinary scientific and cultural significance. To visit Mistaken Point is to walk on surfaces that were seafloor more than 560 million years ago, to look at the ancient sedimented stone and see the impressions of organisms that lived before the first animal with eyes had ever opened those eyes. To stand in the fog and wind that characterize that rugged coastline and try to imagine yourself on the floor of an ancient ocean, surrounded by the fractal fronds of rangiomorphs, in a world of chemical rather than visual signaling, of patient growth rather than active pursuit. The fossils at Mistaken Point are preserved on bedding surfaces, the flat surfaces of ancient rock layers that represent moments in time, snapshots of the ancient seafloor. The most famous of these surfaces covers several square meters and contains dozens of individual fossil specimens of multiple species, all preserved together in the positions they occupied when the volcanic ash cloud that killed and buried them descended from above. This surface has been studied and restudied for decades, with new digital imaging and analysis techniques revealing details that earlier researchers could not have accessed. When you look at the Mistaken Point surface in photographic detail, several things strike you. The first is the sheer density of organisms. This was a crowded seafloor, with organisms packed relatively closely together, competing perhaps for space, certainly for access to the nutrient-rich water above. The second is the variety of forms, different species, different orientations, different sizes, all coexisting on the same patch of seafloor. The third is the evidence of ecological organization, patterns in the distribution of species that are not random, that suggest the existence of genuine ecological structure, of niche differentiation, of the complex web of interactions that characterizes any functioning ecosystem. And the fourth thing that strikes you, if you let yourself feel it rather than just analyze it, is the sheer depth of time that these fossils represent. The organisms captured on this surface lived and died 565 million years ago. The Earth was a profoundly different place. The continents were in different positions, the moon was closer, the day was shorter, perhaps 21 or 22 hours instead of 24. The sun was slightly dimmer, the atmosphere was chemically different, the magnetic field of the Earth was different, everything was different, and yet here is life. Here is organized, complex, ecologically structured life, doing what life does, growing, competing, living and dying in a community, shaping and being shaped by its environment. The fundamental impulse is the same. The details are utterly alien. The combination is one of the most intellectually and emotionally arresting things that science has to offer us. The Flinders Ranges in South Australia are another important Ediacaran locality, and in some ways the most historically significant, since the Ediacaran period takes its name from the Ediacara Hills in this region. The Australian material includes some of the most diverse and best-studied Ediacaran fossils, and it covers a range of environmental settings, from offshore deep water environments to shallow coastal settings. That gives a more complete picture of the range of habitats in which Ediacaran organisms lived. Among the Australian material are some of the most striking and enigmatic Ediacaran organisms. Tribrachidium, for example, has a threefold symmetry, not the bilateral symmetry that characterizes most animals, and not the radial symmetry of modern echinoderms or cnidarians, but a genuine threefold pattern, with three curved lobes arranged around a central point. Tribrachidium has no clear modern relative. Its symmetry pattern is unlike that of any living animal group, and its biological affinity remains uncertain. It is, in the most literal sense, a body plan that left no clear descendants in the modern world. Parvincorina is another puzzling Australian form, small, rounded, with a distinctive anchor-shaped marking on its upper surface. It has been compared to arthropods, the group that includes crabs, insects, and spiders, and to other animal groups, but no comparison is fully convincing. It represents, like so many Ediacaran organisms, a form of life that was genuinely experimenting with something new, and that may or may not have successfully connected to the later mainstream of animal evolution. Spraguina is perhaps the most immediately evocative of the Australian Ediacaran organisms. It has an elongated body with a distinct, somewhat shield-shaped front end and a series of paired lateral lobes running down its body, giving it a superficial resemblance to some early Cambrian arthropods. It has been compared to trilobites, to polychaete worms, and to early relatives of the annelids. The comparison to trilobites is particularly tantalizing because it raises the possibility that Spraguina might represent an ancestor or close relative of one of the most successful groups of early Paleozoic animals. But the comparison is controversial, and many researchers are cautious about it. What all these organisms share, Tribrachidium, Parvincorina, Spraguina, and the dozens of other Australian Ediacaran forms, is a quality of genuine strangeness that resists easy categorization. They are diverse. They show a range of body plans and structural organizations. They include organisms with no obvious modern relatives and organisms that may have very distant but real connections to modern animal groups. Together, they paint a picture of a world in which the basic parameters of complex multicellular life were being explored, tested, and refined through the blind process of evolution, with no predetermined destination, no guarantee of success, no roadmap to the future. The question of how Ediacaran organisms reproduced is one that receives less attention than questions about feeding and behavior, but it is just as fundamental. How did organisms like Dickinsonia or Charnia make more of themselves? How did they spread, colonize new areas of seafloor, establish populations? For sessile organisms like the rangiomorphs, organisms that were attached to the seafloor and could not move to find mates, reproduction through the release of spores or some kind of propagule into the water column makes obvious sense. A rangiomorph could release microscopic reproductive units that drifted with the current and eventually settled on new areas of seafloor, where they would develop into new individuals. This is a strategy used by many modern sessile organisms, including corals and sea fans. There is actually evidence that some rangiomorphs may have reproduced in another way as well, by a process analogous to vegetative reproduction or clonal spreading. At Mistaken Point and other sites, some species are found in spatial patterns that suggest the spread of a genetically identical population from a single original individual, like a ring of mushrooms growing from a single underground mycelial network, or like the clonal spread of a plant through stolons. If rangeomorphs could spread clonally across the seafloor, they might have been able to colonize large areas very efficiently, even without the ability to move. For more mobile organisms like Dickinsonia, the reproductive story is less clear. Some researchers have proposed that Dickinsonia reproduced by simple binary fission, essentially splitting in two, with each half then developing into a new individual. This is a common reproductive strategy in simple animals and in some single-celled organisms. Evidence for this interpretation comes from the observation that some Dickinsonia specimens appear to show asymmetric body proportions consistent with a recent fission event, but the evidence is not conclusive. The question of reproduction is connected to the deeper question of how Ediacaran organisms were organized at the cellular level. Were they truly multicellular in the same sense that modern animals are multicellular, with specialized cell types, with developmental programs that guide the growth of different tissues, with the complex cellular communication that characterizes animal bodies? Or were they multicellular in a simpler sense, aggregations of more or less identical cells, loosely cooperating but not truly specialized? The honest answer is that we do not fully know. The level of cellular organization in Ediacaran organisms is genuinely uncertain, and it may have varied considerably from one group to another. Some Ediacaran organisms may have had relatively simple cellular organization, while others may have been much more complex internally than their simple external appearance suggests. The evolution of cell-type specialization, of the developmental programs that guide the formation of different tissues and organs, is one of the most important and least understood transitions in the history of complex life, and the Ediacaran is the period in which it must have been happening. One of the most philosophically provocative aspects of the Ediacaran is the question of consciousness and experience. We have been talking about organisms that fed, that moved, that reproduced, that, in a functional sense, had lives. But did they experience those lives? Did Dickinsonia, as it slowly moved across the microbial mat, absorbing nutrition from below, have any subjective experience of that process? Did the rangeomorphs swaying in the ancient current have any awareness of their own existence? The question seems almost absurd when applied to creatures so simple and so ancient. And yet it is not a trivial question. The origin of experience, of subjective awareness, of sentience in any form, is one of the deepest unsolved problems in all of science. We know that we are conscious. We have reasonable grounds for believing that many modern animals have significant conscious experiences. But where, in the history of life, did experience begin? What level of neural complexity is required before there is, in the philosopher Thomas Nagel's phrase, something that it is like to be a particular organism? The Ediacaran organisms had no brains. Most of them almost certainly had no nervous system of any kind, no neurons, no synapses, no centralized information processing. The rangeomorphs in particular were almost certainly entirely without any kind of neural tissue. They were complex structures, certainly. They had organization. They responded to their environment in chemical ways. But it seems very unlikely that there was any subjective experience associated with being a rangeomorph in any meaningful sense. With Dickinsonia and Kimberella, the question becomes slightly less clear. If these organisms were animals, as the biochemical evidence suggests, and if they were capable of directed movement and active feeding behavior, then they must have had some mechanism for coordinating that behavior. Whether that mechanism involved anything like a nervous system, even a very simple one, is genuinely uncertain. The preservation of Ediacaran soft tissue fossils is not good enough to show us the internal organization of these organisms with the clarity that would be needed to answer the question definitively, but the question itself is worth sitting with. The Ediacaran is the period in which the biological substrate for experience, if experience has a biological substrate in the way most scientists believe it does, was beginning to be assembled. The first nervous systems were not yet present. The first complex behaviors were just beginning to emerge. We are watching, from our distant vantage point hundreds of millions of years later, the very early stages of the process by which the universe began, slowly and tentatively, to know itself through the experience of living creatures. That is not a trivial thing. That is, arguably, one of the most important processes that has ever occurred on this planet, and the Ediacaran is where it began. Let us return now to the ecology of the Ediacaran world, and think about some of the more subtle and complex dynamics that were playing out on those ancient seafloors. Because the Ediacaran was not just a world of organisms growing passively in the current. It was a world of ecological relationships, of competition, of facilitation, of the complex web of interactions that makes any ecosystem more than the sum of its parts. Competition for space on the seafloor was certainly a significant ecological force. The rangiomorphs needed to attach to the substrate. Organisms like Dickinsonia needed access to undisturbed microbial mat. Kimberella needed mat surface to graze. With multiple species competing for the same seafloor real estate, there would have been competitive exclusion, situations where one species outcompeted another for a particular patch of substrate, and competitive coexistence, situations where species divided up the available space in ways that allowed multiple species to persist. The analysis of spatial patterns in Ediacaran fossil communities has been used to try to understand these competitive dynamics. At Mistaken Point, for example, detailed statistical analysis of the positions of different species has revealed patterns consistent with both competition and facilitation, situations where the presence of one species appears to create conditions favorable for another, and situations where species appear to exclude one another from their immediate vicinity. This is genuine community ecology. This is the study of how organisms interact with one another and with their shared environment, and it is being applied to organisms that lived more than 500 million years ago. The fact that we can do this, the fact that the preservation at sites like Mistaken Point is good enough to allow this kind of analysis, is a remarkable scientific achievement, made possible by the combination of exceptional fossil preservation and modern analytical techniques. Facilitation in particular is an interesting concept in the Ediacaran context. In modern ecosystems, facilitation, where one organism creates conditions that benefit another, is an important ecological process. Kelp forests create habitat for dozens of other species. Beavers create wetlands that support entire communities. Elephant paths open clearings that other species exploit. If facilitation was operating in Ediacaran communities, it would suggest that these ancient ecosystems had a level of ecological complexity far beyond what we might naively expect from such early, simple-seeming organisms. And there is reason to think it was. The spatial analyses of Ediacaran communities consistently reveal non-random patterns that are most parsimoniously explained by ecological interactions, including facilitation. The Ediacaran world was not just a collection of independent organisms doing their own thing on the seafloor. It was a community, in the full ecological sense of the word, a system of interacting species whose collective behavior was more than the sum of its individual parts. The question of what Ediacaran organisms looked like in life truly looked like, with the texture and color and three-dimensional presence that a living organism has, is one that science cannot fully answer. We have impressions in rock, we have chemical signatures, we have the shapes and proportions of bodies preserved in sediment, but we do not have color, we do not have texture beyond what the rock preserves, we do not have the shimmer of life in them. Artistic reconstructions of Ediacaran organisms have become increasingly sophisticated over recent decades as our scientific understanding of these creatures has deepened. Modern paleoartists work closely with paleontologists to create images that are as scientifically defensible as possible, incorporating the latest research on body plans, ecological context, and environmental setting. These reconstructions typically show the Ediacaran world as a largely colorless or muted color environment, the microbial mat a dark greenish or brownish carpet, the organisms themselves perhaps pale or translucent, since many of them may have lacked the pigmentation that modern animals use for signaling, camouflage, and mate selection. Without eyes, there was no need for vivid color. Color as a visual signal is only useful if there are eyes to perceive it. The Ediacaran was a world before vision, and its aesthetics were therefore fundamentally different from anything we can directly experience. But colorless does not mean lifeless in any aesthetic sense. The Ediacaran world had its own beauty, the fractal elegance of the rangiomorphs, the quiet regularity of Dickinsonia's bilateral modules, the gentle sway of fronds in the ancient current. It was a world of form rather than color, of structure rather than behavior, at least in its early phases. As the period progressed and organisms became more active, more mobile, more behaviorally complex, the aesthetic character of the world must have shifted somewhat toward movement and interaction. But even at its most active, the Ediacaran remained a world profoundly different in character from anything that came after. There was no sound in the Ediacaran Ocean, or rather there was sound, the sound of water moving and sediment shifting, but there was no sound produced by living organisms. No communication through sound, no calls, no clicks, no songs. Sound as a biological signal, sound as language, even in the most primitive sense, would have to wait for later, more complex organisms. The Ediacaran communicated, if it communicated at all, through chemistry. Through the release of dissolved molecules into the water. Through contact chemistry, perhaps, the way cells communicate with one another through chemical signals at the surface. This chemical communication, if it existed in any meaningful way, was another invisible feature of the Ediacaran world, another dimension of its existence that we can barely access from our distant vantage point. Another reminder of how different life was in this ancient chapter from anything we can directly observe or experience. As we move deeper into the later Ediacaran, we see the community changing. Not suddenly, not catastrophically, at least not in the first instance, but gradually, systemically, in ways that reflect the ongoing processes of evolution and ecological change. One of the most significant of these changes is the gradual increase in the diversity and abundance of trace fossils, of the tracks, trails, and burrows left by organisms moving through the sediment. In the early Ediacaran, trace fossils are essentially absent. The seafloor was undisturbed, the microbial mat was continuous, and whatever organisms were moving on the surface were not leaving traces that preserved well. But in the later Ediacaran, from about 550 million years ago onward, trace fossils become increasingly common and increasingly complex. The earliest trace fossils in the Ediacaran record are simple horizontal trails at or near the sediment surface, consistent with small organisms moving across the top of the microbial mat. There is no penetration of the sediment, no burrowing into the mat, no vertical mixing. But over time, the traces become more complex. They become deeper, they begin to show evidence of organisms moving through the sediment rather than just across it. This progression from simple surface trails to more complex burrowing is not just a biological development, it is an ecological revolution. When organisms start burrowing into the sediment, they begin to change the physical structure and chemical environment of the seafloor in ways that have cascading consequences. They mix the sediment, disrupting the layered structure that the microbial mat depends on. They introduce oxygen into layers of sediment that were previously anoxic. They change the distribution of nutrients through the sediment. They alter the conditions for every other organism living in the same habitat. This process, the bioturbation of the seafloor by burrowing organisms, is sometimes described as one of the most significant ecological changes in the history of life, and it began in the Ediacaran. It was not the result of any single organism or any single innovation. It was a gradual process, playing out over tens of millions of years, as the evolutionary capability for burrowing slowly developed and as the ecological consequences of burrowing slowly accumulated. The organisms responsible for the earliest Ediacaran burrows are not well known. The burrows themselves are preserved, but the organisms that made them rarely are. They were presumably small, soft-bodied, bilaterally symmetrical animals capable of directional movement, the kind of organisms that would eventually give rise to the diverse fauna of burrowing worms and early animals that characterize the Cambrian. But their precise identity and evolutionary relationships remain uncertain. What we know is that they were there, in the later Ediacaran, beginning to transform the seafloor, beginning to set in motion the process that would ultimately destabilize the microbial mat world that had sustained Ediacaran complex life, beginning, inadvertently, to undermine the very ecological foundation on which the Ediacaran world was built. There is a famous thought experiment in evolutionary biology sometimes called replaying the tape of life. The question is, if you could go back to some early point in the history of life and replay evolution from that point forward, would you get the same result? Would you get organisms like us? Would you get intelligence, consciousness, technological civilization? Or would the inherent contingency of evolution, the role of chance, of random mutation, of the particular accidents of environmental history, produce something completely different? The Ediacaran makes this thought experiment particularly vivid, because the Ediacaran is a world in which the outcome was genuinely uncertain. When we look at the Ediacaran, we are not looking at an inevitable march toward the world we know. We are looking at a genuine experiment in progress with multiple possible outcomes. Some of the body plans being tried out in the Ediacaran led nowhere. They were evolutionary dead ends, experiments that did not persist. Others were the roots of the great lineages of animals that have dominated Earth ever since. But at the time, from within the Ediacaran world, there was no way to know which was which. Dickinsonia, as we have noted, appears to have been an animal biochemically, but it left no clear descendants. Its distinctive body plan, the bilateral modules, the flat, quilted surface, the mode of feeding by absorption from below, appears to have been discontinued. No modern animal looks like Dickinsonia. No modern animal feeds quite the way Dickinsonia appears to have fed. It was an experiment that ran for tens of millions of years, achieved a remarkable degree of success and diversity by the standards of its time, and then ended. The rangiomorphs are even more striking in this regard. If they represent a genuinely separate experiment in multicellular organization, an alternative approach to building a large, complex body that was tried and ultimately abandoned, then they are the most dramatic example in the entire fossil record of evolutionary innovation that did not persist. An entire approach to multicellular life, an entire set of strategies for absorbing nutrients and growing complex forms. An entire ecological role filled by organisms for tens of millions of years, and then gone, or almost gone, because there is an argument made by some researchers that the apparent disappearance of the rangiomorphs and other Ediacaran organisms is partly an artifact of preservation. The organisms of the Ediacaran preserved because they lived on the seafloor and were buried by volcanic ash or rapid sediment deposition. If their descendants became smaller or more mobile or changed their ecological habits in ways that made them less likely to be preserved, we might simply not see them in the later fossil record. They might still be there, hidden in the gaps of the fossil record, connecting the Ediacaran to the Cambrian in ways we have not yet been able to detect. This is a genuine scientific possibility. The Cambrian explosion, the rapid diversification of animal body plans in the early Cambrian, beginning around 538 million years ago, is in some sense the successor to the Ediacaran. The organisms of the Cambrian did not appear from nowhere. They had ancestors. Those ancestors were somewhere in the late Ediacaran world, evolving, diversifying, preparing for what was to come. But identifying those ancestors in the fossil record is extraordinarily difficult, and the connection between Ediacaran and Cambrian faunas remains one of the great open questions in evolutionary biology. There is another dimension of the Ediacaran world that we have not yet fully explored, and it is one that tends to be underappreciated even in scientific discussions of the period. That is the dimension of ecological engineering, the ways in which Ediacaran organisms, through their mere existence and their biological activities, were changing their own environment. We have already discussed the bioturbation of the seafloor by burrowing organisms in the later Ediacaran, but there were other ways in which the Ediacaran organisms were transforming their world. The rangiomorphs, for example, by growing upright from the seafloor, were creating physical structures in the water, structures that would have altered the local flow of current, creating sheltered zones downstream and turbulent zones upstream, changing the distribution of nutrients and particles in the water. This is habitat engineering, the creation by organisms of physical structures that alter the local environment and affect other organisms. Modern examples of habitat engineering are everywhere: coral reefs, oyster beds, kelp forests, mangroves, termite mounds. In every case, the engineering organisms create structures that affect not just themselves, but the entire community of organisms around them. If the rangiomorphs were habitat engineers, and their upright, frond-like forms certainly suggest that they were creating physical structures that would have had hydrodynamic effects, then the communities at places like Mistaken Point were already experiencing a level of ecological complexity driven by habitat engineering that goes significantly beyond simple resource competition. The implications of this are interesting. If rangiomorphs were habitat engineers, then the structure of the Ediacaran community was partly self-reinforcing. The presence of rangiomorphs created conditions that were favorable for other rangiomorphs and potentially for other organisms as well. This kind of positive feedback in ecosystem structure is a hallmark of complex, mature ecosystems, and finding evidence of it in the Ediacaran suggests that ecological complexity was established remarkably early in the history of complex multicellular life. By the time we reach the last 20 or 30 million years of the Ediacaran, roughly from around 550 to 538 million years ago, the world is visibly changing. The fossil communities of this period, found in places like the Nama group in Namibia, look different from the earlier Ediacaran communities at Mistaken Point or in the early White Sea assemblages. Some Ediacaran forms that were abundant earlier are becoming rare or absent. New forms are appearing, organisms with more clearly defined bilateral symmetry, organisms with harder body parts, organisms that were more actively mobile. The trace fossil record is becoming richer and more complex. The chemical evidence from the rocks suggests that the ocean chemistry was changing, that oxygen levels were fluctuating, that the chemistry of the seafloor sediment was being altered by increased biological activity. The Ediacaran world was in transition. The stable, mat-dominated seafloor communities of the early and middle Ediacaran were being destabilized. The ecological relationships that had sustained those communities were being disrupted. New ecological relationships were forming, harder, faster, more energetic in every sense than what had come before, and from within the old world, the new world was emerging. In the cracks and margins of the Ediacaran ecosystem, in the populations of small, active, burrowing bolithurians that were transforming the seafloor, in the descendants of organisms like Kimberella that were beginning to develop harder structures and more complex behaviors, in all of these places, the Cambrian world was being assembled, not explosively, not suddenly. The Cambrian explosion is in many ways a misnomer. It was not an explosion by human standards, but a diversification that played out over millions of years. But it was a transformation, a genuine, fundamental change in the character of life on Earth. The world of the rangiomorphs was giving way to the world of the trilobites. The world of passive absorption was giving way to the world of active predation. The world of the microbial mat was giving way to the world of the bioturbated, biologically complex seafloor that we still live in today. The Ediacaran was ending. Not in a day, not in a year, not even in a million years, but over the course of tens of millions of years, the world that had made the Ediacaran organisms possible was becoming a world that could no longer sustain them. And what would rise from the transformation would be something genuinely new, something that built on the evolutionary foundations laid down in the Ediacaran, but that took them in directions that the Ediacaran organisms themselves could never have reached. This is where we will pause at the threshold of the Great Transition, the ending of the Ediacaran world, and the beginning of everything we recognize as the mainstream of animal life. We have walked the ancient seafloors. We have met Charnia and Dickinsonia and Kimberella and Cloudina and Tribrachidium and all the other strange inhabitants of this forgotten world. We have tried to understand the ecology and the chemistry and the evolutionary dynamics of a world more than 500 million years old. And we have found, I hope, that this world, alien as it is, unfamiliar as it is, resistant as it is to our normal categories and expectations, was a real world, a functioning world. A world with its own depth and complexity and, in some sense, its own dignity. In the second part of this documentary, we will follow the ending of that world, and we will explore what its ending meant for everything that came after. We will sit with the slow, systemic unraveling of the Ediacaran ecosystem. We will watch the Cambrian world rise from the ruins of the old. And we will reflect on what the Ediacaran means for us, for how we understand life's history, for how we think about complexity and evolution and the deep time of this ancient planet. But for now, let us stay a moment longer in the Ediacaran world. Let us stand on the ancient seafloor in the cold, dark water and look at the fractal fronds of the rangiomorph swaying in the current, at the slow, purposeful movement of Dickinsonia across the microbial mat, at the quiet, patient life of these extraordinary organisms going about the business of being alive in a world that had not yet learned to be violent, had not yet learned to be fast, had not yet learned to be complex in the way that everything after it would be. There is something valuable in that quiet, something worth paying attention to, something that the frenetic complexity of the modern world with its constant noise and motion and competition makes it easy to forget. Life began slowly, life began quietly, life began with patience and with the kind of deep geological time that we can barely imagine. And it was in that slow, patient beginning in the Ediacaran, in the world before eyes, before teeth, before pursuit, that the foundations for everything we are were laid. This has been part one of our exploration of the Ediacaran period. We have covered the origins of the period, the nature of its organisms, the ecology of its communities, and the first signs of the transformation that would eventually bring the Ediacaran world to its close. In part 2, we will continue this journey, following the ending of the Ediacaran, exploring the transition to the Cambrian, and reflecting on the deeper meaning of this extraordinary chapter in Earth's history. Thank you for watching. Please let us know in the comments where you are watching from and what time it is there. Every comment reminds us of the remarkable fact that people around the world, separated by thousands of miles and entirely different daily lives, can come together around the story of ancient life, can share, for a moment, the experience of looking back across 500 million years of time and trying to understand what was there. Please subscribe to the channel if you have not already done so. More videos on the deep history of life on Earth are coming, and each one of them is a journey into the extraordinary, improbable, endlessly fascinating story of life on this planet. We left the Ediacaran seafloor at the edge of a great transformation. The rangeomorphs were still swaying in the ancient current. Dickinsonia was still moving slowly across its microbial carpet. Kimberella was still rasping at the matte surface with its feeding organ, but something was changing. Something in the chemistry of the ocean, something in the structure of the seafloor, something in the evolutionary trajectories of the small, active, burrowing organisms that were beginning to transform the world from below. The Ediacaran was ending, and in its ending, everything we know was beginning. We return now to the ancient world. We return to time more than 500 million years ago, to a planet that was already 4 billion years old, but that was, in the most important biological sense, still very young. We return to the Ediacaran, to the world before eyes, before shells, before the great diversification of animal forms that would eventually produce everything from the first fish to the last human being. In part one of this documentary, we walked the ancient seafloors and met the inhabitants of the Ediacaran world. We met the fractal rangeomorphs of Mistaken Point, those extraordinary organisms whose self-similar branching structures maximized surface area for nutrient absorption in ways that have no real equivalent in the modern world. We met Dickinsonia, the bilateral quilted organism that moved slowly across the microbial mat, absorbing nutrition from below, leaving behind it patches of disturbed and depleted mat surface. We met Kimberella with its rasping feeding organ, its directed movement, its possible status as an early relative of the mollusks. We met Cloudina, one of the first organisms in Earth's history to construct a mineralized shell, a thin, fragile nested cone structure that nevertheless represented one of the most consequential biological innovations in the history of life. We explored the ecology of the Ediacaran, the mat-dominated seafloor communities, the competitive and facilitative interactions between species, the evidence for genuine ecological organization and structure in communities more than 565 million years old. We explored the chemistry of the ancient ocean and the way that chemistry shaped and constrained the possibilities of early complex life. And we stood at the threshold of the great transformation that would eventually bring the Ediacaran world to its close. Now we cross that threshold. Now we follow the Ediacaran into its ending, and we ask the questions that its ending raises. Why did this world disappear? What replaced it, and why? What was lost when the last rangeomorph ceased to grow, and the last Ediacaran mat community was buried and compressed into stone? And what was gained? What was the foundation that the Ediacaran laid for everything that followed? These are not simple questions. They do not have simple answers. But they are among the most important questions we can ask about the history of life on Earth, because they go to the heart of what life is, how it changes, and what it means for something as complex and as contingent as the living world to transform itself from one state into another. Let us begin. To understand the ending of the Ediacaran, we first need to understand what made it stable. What were the conditions that allowed the Ediacaran world to persist in essentially the same basic ecological mode for so many tens of millions of years? Because the Ediacaran was not a world in constant flux. For much of its duration, it was a world of remarkable stability, a world in which the same basic ecological relationships, the same fundamental strategies for making a living, persisted for periods of time that make human history look like the blink of an eye. The foundation of that stability, as we discussed in Part 1, was the microbial mat. The mat was not just a food source or a substrate. It was the organizing principle of the entire Ediacaran benthic ecosystem, the ecological anchor around which everything else was organized. And the mat was itself stable because of specific physical and chemical conditions that characterized the Ediacaran seafloor. Chief among those conditions was the relative absence of bioturbation. In the modern ocean, the top layer of seafloor sediment is constantly mixed and reworked by the burrowing activities of thousands of species of worms, mollusks, crustaceans, echinoderms, and other organisms. This biological mixing, bioturbation, happens continuously and pervasively, preventing the development of stable layered structures in the sediment and maintaining the chemical conditions of the sediment in a well-mixed, relatively oxygenated state. In the Ediacaran, none of this was happening. The organisms capable of significant burrowing were absent or extremely rare. The seafloor was, by modern standards, essentially undisturbed, and this undisturbed condition allowed the microbial mat to develop in the way that it did, in continuous, stable, layered sheets that maintained specific chemical gradients across their thickness. The mat was chemically stratified. At the surface, where light could penetrate, photosynthetic organisms dominated. Just below the surface, in the slightly darker zone, different metabolic strategies prevailed. Deeper still, in the anoxic layers of the sediment below the mat, yet another set of chemical processes, sulfate reduction, methane production, were operating. This chemical stratification was delicate. It depended on the stability of the mat itself, on the maintenance of specific gradients of oxygen and organic carbon and other chemical species across the thickness of the mat and the sediment. When those gradients were disrupted, when the mat was physically mixed or the sediment was bioturbated, the chemical system of the mat collapsed. The organisms that had depended on the mat's specific chemical environment found themselves in an environment that was fundamentally different, one that their evolutionary history had not prepared them for. This is the key to understanding the ending of the Ediacaran. It was not primarily a story of one set of organisms directly outcompeting another. It was a story of environmental transformation, of the Ediacaran ecosystem gradually being undermined by the biological activities of the organisms that were evolving within and around it, until the physical and chemical conditions that had sustained the Ediacaran community no longer existed. The Ediacaran world was, in a very real sense, eaten from the bottom up. Not by predators or not primarily by predators, though predation played a role, but by the slow, relentless biological transformation of the seafloor itself. Let us trace this transformation more carefully. Let us follow it through time, from the first tentative signs of bioturbation in the later Ediacaran to the fully bioturbated seafloor of the early Cambrian, and try to understand the cascade of consequences that this physical change produced. Around 555 million years ago, in the later Ediacaran, the trace fossil record begins to show clear evidence of organisms moving through the sediment rather than just across its surface. These early traces are not deep. They penetrate only a centimeter or two into the mat, and they are simple in structure, without the complex branching or backfilling behavior that characterizes later, more sophisticated burrowers. But they are there. Small animals, their identity uncertain, their bodies soft and largely unpreserved, were beginning to explore the sediment itself as a habitat. What were they looking for? The most likely answer is food. The microbial mat was rich in organic carbon. An organism that could burrow into the mat and process the microbial material within it would have access to a food source that was not being exploited by the surface-dwelling Ediacaran community. The niche was open, the reward was substantial, and evolution, given enough time and enough variation, will find any niche that is energetically rewarding. The early burrowers were pioneers into an unexploited ecological space. They were doing something genuinely new, moving through the sediment, processing mat material internally, leaving behind them altered sediment with different chemical properties from the undisturbed material around it. And in doing so, they were, inadvertently, beginning to change the conditions of their own habitat and the habitats of the organisms around them. The changes were initially minor. A few small burrows in a vast expanse of undisturbed mat are not going to transform the chemistry of the seafloor, but burrowing, once it starts, tends to be self-reinforcing. A burrowing organism that successfully exploits the organic-rich sediment will reproduce. Its descendants will burrow too. Mutations that improve burrowing efficiency will be selected for. Over thousands of generations, which for small, short-lived animals might represent only tens of thousands of years, the burrowing capability becomes more sophisticated, the burrows deeper, the chemical impact more substantial. And as the impact on the sediment becomes more substantial, the chemical gradients that maintain the microbial mat begin to shift. Oxygen from the water above penetrates deeper into the sediment along the walls of burrows. The chemically reduced zones that certain mat organisms depend on become smaller and more disrupted. The organic carbon that would otherwise accumulate in the sediment and be processed by the mat community is instead being ingested and processed by the burrowers. The mat begins to thin. In places, it begins to fragment. This is not a sudden collapse, it is a slow erosion. The mat does not disappear everywhere at once. It persists in patches, in environments where burrowing is less effective, in areas where the physical conditions, current strength, sediment grain size, water chemistry are less favorable for burrowers. But the trend is clear and it is irreversible. Once burrowing begins in earnest, the Ediacaran mat world is on a trajectory toward eventual disappearance. And as the mat world changes, the organisms that depended on it change too. Or rather, many of them do not change fast enough. The evolutionary responses available to organisms like the rangiomorphs, with their passive absorption strategy and their sessile, attached lifestyle, were limited. They could not suddenly develop the ability to burrow. They could not suddenly start predating. They were committed by their body plans and their evolutionary history to a particular way of life, a way of life that worked beautifully in a world of stable, abundant microbial mat, and that worked less and less well as that mat world was eroded. This is one of the most poignant aspects of the Ediacaran ending. The organisms that were most successful in the Ediacaran world, the rangiomorphs, with their elegant fractal forms and their efficient passive absorption, were in a sense too specialized. They were too perfectly adapted to a particular set of conditions, and those conditions were changing. The very success that had made them abundant and diverse in the Ediacaran world was, paradoxically, a constraint that prevented them from adapting to the world that was replacing it. The concept of evolutionary constraint is important here and worth pausing to explore. Evolutionary constraint refers to the way in which an organism's existing body plan and biological organization can limit the range of evolutionary directions available to it. An organism that has been highly optimized for one particular way of life, one particular set of environmental conditions, one particular ecological role, may find it very difficult to evolve toward a radically different lifestyle, even if that different lifestyle would be advantageous in the new conditions it finds itself in. The rangiomorphs exemplify this. Their fractal body plan was extraordinarily well suited for passive osmotic absorption in a nutrient-rich, stable marine environment. The same body plan was not well suited for active locomotion, for predation, for burrowing, or for any of the other ecological strategies that were becoming increasingly important as the Ediacaran world transformed. And the evolutionary distance between a fractal, sessile absorber and any of those more active lifestyles was enormous, too enormous, apparently, to be bridged by the incremental process of natural selection in the time available. This is not a failure of the rangiomorphs in any meaningful sense. It is simply the reality of evolution, that adaptation to current conditions can make adaptation to future conditions more difficult, that the very process of becoming good at something constrains your ability to become good at something very different. The rangiomorphs were extraordinarily good at being what they were. The problem was not with them. The problem was with the world. The world was changing faster than they could keep up with it. Or more precisely and more fairly, the world was changing in a direction that they could not follow. Some Ediacaran organisms could follow it, or at least approximate it. Some of the more mobile, more behaviorally complex Ediacaran organisms, the bilaterians that were leaving the early trace fossils, the organisms that were beginning to develop hard parts in response to predation pressure, these were the organisms that could adapt to the changing world. These were the evolutionary lines that would carry the lessons of the Ediacaran into the Cambrian and beyond, but the rangiomorphs were not among them, and neither, it appears, were most of the organisms that gave the Ediacaran its distinctive ecological character. Let us talk about predation more explicitly, because it is the other major force, alongside bioturbation, that was transforming the Ediacaran world in its final stages. We mentioned in Part 1 that Cloudina, one of the first mineralized organisms, showed evidence of predatory drilling, holes bored through its shell by some unknown predator. This evidence is deeply significant because it tells us that by the end of the Ediacaran, predation was not just beginning to emerge as an ecological force, but was already sophisticated enough to have driven the evolution of defensive mineralized structures. Think about what that means. For predatory drilling to be evidenced in the fossil record, you need a predator that has evolved the behavioral and physical capability to actively target a shelled prey organism, to apply sustained mechanical force to break through that shell, and to extract the soft body within. This is not the behavior of a simple, early predator. This is the behavior of an organism with relatively complex feeding behavior, with the sensory capability to locate and assess prey, with the physical tools to overcome defensive structures. The predators of the late Ediacaran are almost entirely invisible to us. Their soft bodies left no record, and even the traces they left are ambiguous, but their effects are written in the biology of their prey. The evolution of Cloudina's shell and the bore holes that penetrate it tell us that predation in the late Ediacaran was a real and significant ecological force, creating strong selection pressure for defensive innovations. And predation does not just affect the specific organisms being preyed upon. Predation transforms entire ecosystems. It creates cascades of ecological effects that ripple through every level of the food web. When a significant predator appears in an ecosystem, every organism in that ecosystem must adapt to it by developing defenses, by changing its behavior, by moving to different habitats, by evolving faster or smaller or more camouflaged or more toxic. The arrival of significant predation in the Ediacaran world would have been, in ecological terms, a seismic event. For the Ediacaran organisms that had no shells, no fast locomotion, no behavioral defenses, and no evolutionary history of dealing with predators, which is to say, most of the classic Ediacaran fauna, the arrival of predation was catastrophic. Not instantly catastrophic, evolution does not work on instant timescales, but catastrophic in the sense that the ecological conditions that had previously been tolerable were now intolerable. The energy budget of an Ediacaran organism now had to account for the possibility of being eaten, and for organisms whose entire body plan was predicated on passive absorption in a safe environment, this new accounting did not balance. The emergence of predation in the late Ediacaran was, in this sense, not just a biological event, but an ecological revolution. It changed the fundamental terms of existence for every organism in the affected communities. It created a world in which being invisible, being protected, being fast, being able to sense danger and respond to it, suddenly had survival value that they had lacked in the quieter Ediacaran communities of earlier times. And it was this new world, this world of predation and counterpredation, of arms races between offense and defense, of the constant evolutionary pressure created by organisms trying to eat each other, that would define the Cambrian and all the periods that followed. The world that we live in, the world that produced us. The transition from the Ediacaran to the Cambrian has sometimes been described as one of the most dramatic events in the history of life. The Cambrian explosion, the rapid diversification of animal body plans that occurred beginning around 538 million years ago, is one of the most studied and most debated events in paleontology. Darwin himself was troubled by it because it seemed to suggest that complex animal forms appeared suddenly in the fossil record without clear ancestors, which contradicted his theory of gradual evolution through natural selection. We now know that Darwin's concern, while understandable given the state of knowledge in his time, was misplaced. The Cambrian explosion was not sudden in any geologically meaningful sense. It played out over tens of millions of years. And its apparent suddenness in the fossil record is partly an artifact of the much better preservation of hard-shelled organisms in Cambrian and later rocks compared to the soft-bodied Ediacaran organisms. Once organisms evolved mineralized skeletons, their preservation potential in the fossil record increased enormously, giving the impression of a sudden appearance even though the underlying biological lineages had been evolving throughout the Ediacaran. But even accounting for these taphonomic biases, even recognizing that the fossil record is an imperfect and biased sample of ancient biodiversity, the Cambrian does appear to represent a genuine and significant acceleration in the pace of evolutionary diversification. Something happened in the early Cambrian that drove an unusually rapid proliferation of new body plans and ecological strategies. And understanding what that something was requires understanding the Ediacaran world that immediately preceded it. The most convincing explanation for the Cambrian explosion is ecological. Specifically, it is the cascading ecological consequences of the arrival of predation in the late Ediacaran. Predation, as we have discussed, creates selection pressure for defensive innovations, shells, spines, hard carapaces, the ability to burrow and hide in the sediment. These defensive innovations, in turn, create new selection pressure on predators to evolve better sensory systems for detecting hidden prey, better physical tools for breaking through shells, better behavioral strategies for hunting in a world where prey is better defended. This is the predator-prey arms race, and once it starts, it tends to accelerate. Each innovation on one side of the arms race creates pressure for a counter-innovation on the other side. Shells drive better drilling capability. Better drilling capability drives thicker shells. Thicker shells drive more powerful drilling or more sophisticated approaches, poison, entrapment, overwhelming force. More sophisticated predation drives better camouflage, faster escape, more effective burrowing. And so on in a self-reinforcing cycle of evolutionary escalation that once started tends to produce an explosion of morphological diversity in a relatively short period of geological time. The Cambrian explosion in this view is the result of a biological arms race that began in the Ediacaran, that began specifically with the arrival of the first significant predators and the first defensive responses. Cloudina's shell was not just a response to local predation pressure. It was the first shot in a war that would last for 500 million years, and that would produce, in the process, the extraordinary diversity of animal forms that has characterized Earth ever since. Let us spend some time with the organisms of the early Cambrian, the immediate successors of the Ediacaran world, because in doing so we can see both the continuities and the discontinuities between the Ediacaran and what came after. The Cambrian fossil record is remarkable for its diversity and for the quality of its preservation at certain sites. The Burgess Shale in British Columbia is perhaps the most famous, a site discovered in the early 20th century by Charles Walcott and subsequently restudied to reveal an extraordinary array of soft-bodied animals preserved in exceptional detail. The Chengjiang biota in China is another extraordinary site, slightly older than the Burgess Shale, preserving an even more primitive assemblage of early Cambrian animals. When you look at the organisms of the Burgess Shale and Chengjiang, the contrast with the Ediacaran is immediately striking. These are recognizable animals, animals with heads and tails, with sensory organs including eyes, with limbs, with guts, with complex behaviors. Anomalocaris, one of the largest Cambrian predators, was a sophisticated hunting machine, with large compound eyes on stalks, grasping appendages for capturing prey, and a circular, jaw-like mouth lined with hard plates for crushing shelled prey. It was a predator of a sophistication that had no precedent anywhere in the Ediacaran record. Trilobites, the iconic, three-lobed arthropods that dominate so much of the early Paleozoic fossil record, had complex compound eyes, hard exoskeletons, jointed legs, and behavioral repertoires that included the ability to roll into a ball for defense, suggesting that the predation they were defending against was both real and sophisticated. Hallucigenia, the bizarre organism with spines on one side and tentacles on the other, long a source of confusion for paleontologists before its correct orientation was finally established, was walking on the seafloor on paired, clawed legs, a level of locomotor complexity that has no real parallel in the Ediacaran record. The Cambrian world was faster, louder in a metaphorical sense, more violent, more energetically demanding, more ecologically complex by almost any measure. And it was operating on a fundamentally different physical substrate, a bioturbated seafloor, mixed and reworked by the burrowing activities of hundreds of species, a seafloor without the stable microbial mats that had defined the Ediacaran benthic environment. The transition from the Ediacaran to the Cambrian, seen in these terms, was not just a change in the organisms occupying the ocean. It was a change in the physical character of the ocean floor. It was a change in the fundamental terms of ecological existence, from a world organized around stability and passive absorption to a world organized around competition, predation, and active exploitation of resources. From a world of patience to a world of speed. But let us not be too quick to dismiss the Ediacaran as merely a prelude. Let us not fall into the trap of seeing the Ediacaran purely in terms of what came after, of judging it by the standards of the Cambrian and finding it wanting. The Ediacaran was not an inferior version of the Cambrian. It was a different kind of world with its own standards, its own logic, its own ecological completeness. The philosopher of science would say that the Ediacaran world was well adapted to its conditions. The rangiomorphs were not failures because they could not compete with Cambrian arthropods. They were extraordinary successes in the world they actually lived in, the world of the stable mat community, the world of passive nutrient absorption, the world of fractal elegance and chemical simplicity. They achieved in that world levels of diversity and abundance and ecological complexity that represent genuine biological success by any reasonable standard. The question is not why the Ediacaran organisms were not as good as Cambrian animals. The question is why the conditions that had sustained Ediacaran life for tens of millions of years eventually ceased to exist. And the answer to that question, as we have seen, is biological. The organisms of the Ediacaran world were themselves part of the process that transformed it. The evolutionary innovations that arose within and around the Ediacaran community, burrowing, predation, mineralized hard parts, were not external forces invading from outside. They were the products of the same evolutionary process that had produced the Ediacaran organisms in the first place. They were the next chapter in the same story. Life changes the world, and the changed world changes life. This is the principle that runs through the entire history of life on Earth, and it runs through the Ediacaran transition with particular clarity. The cyanobacteria oxygenated the atmosphere and made complex eukaryotic life possible. The Ediacaran organisms pioneered multicellular body plans and ecological organization and made the Cambrian diversification possible. The Cambrian organisms transformed the seafloor and the ecology of the ocean and made all subsequent animal evolution possible. And so on, through 500 million more years of biological history, each chapter building on the last, each transformation creating the conditions for the next. The Ediacaran is not separate from this story. It is not a dead-end appendage. It is one of the most important chapters in the story, perhaps the most important chapter, in the sense that it established the fundamental biological innovations on which everything else was built. What were those fundamental innovations? What did the Ediacaran contribute to the long-term history of life, beyond the specific organisms that characterized it? The first and most fundamental contribution was multicellularity itself, not just the existence of multiple cells in the same body, but the coordination of those cells into functional units, the specialization of cells for different purposes, the development of the communication systems that allow a multicellular body to function as a coherent whole rather than as a collection of individual cells doing their own thing. Multicellularity was not invented in the Ediacaran. Single-celled organisms had been forming loose multicellular associations for hundreds of millions of years before the Ediacaran began. But complex, integrated, functionally differentiated multicellularity, the kind of multicellularity that allows an organism to have something like tissues, something like organs, something like a body plan, this appears to have been an Ediacaran innovation, or at least to have reached its first flowering in the Ediacaran. The developmental programs that guide the growth of a multicellular body from a single fertilized cell, the genetic regulatory networks that tell each cell what type to become, where to position itself in the body, what to produce and what to respond to, are among the most complex and important products of evolution. These programs, in their most basic outlines, appear to have been assembled during the Ediacaran, in the context of the simple but genuinely multicellular organisms that characterized the period. When the Cambrian animals diversified so rapidly and so dramatically, they were not inventing these developmental programs from scratch. They were elaborating, modifying, combining, and extending programs that had been developed in the Ediacaran. The genetic toolkit of animal development, the Hox genes, the signaling pathways, the transcription factor networks that guide the development of animal bodies, was assembled in its basic form before the Cambrian. The Cambrian explosion was, in part, an explosion of the ways that toolkit could be deployed, not an invention of the toolkit itself. The second fundamental contribution of the Ediacaran was bilateral symmetry. We have discussed this in relation to specific organisms like Dickinsonia and Kimberella, but it is worth pausing to appreciate just how consequential bilateral symmetry was as an evolutionary innovation. Bilateral symmetry, having a left side and a right side that are mirror images, with a definite front end and back end, is so fundamental to the body plans of most modern animals that we rarely stop to appreciate it as an evolutionary invention. But it was an invention. The first organisms were radially symmetrical or asymmetrical. Their bodies had no preferred direction of movement, no concentrated sensory apparatus at one end, no distinction between the part that encounters the environment first and the part that follows behind. Bilateral symmetry changed all of this. An organism with bilateral symmetry can move in a preferred direction forward toward its front end, where sensory organs are concentrated. The concentration of sensory organs at the front end, at the leading end, the end that encounters the environment first, created selection pressure for the further elaboration of those organs, for the development of more sophisticated sensory capabilities, for the centralization of nervous tissue near the sensory organs. This centralization of nervous tissue is the origin of the brain, or rather of the proto-brain structures that evolution would eventually elaborate into the complex central nervous systems of vertebrates and many invertebrates. Bilateral symmetry, in other words, is the evolutionary prerequisite for directed movement, for complex sensory systems, for centralized nervous systems, and ultimately for brains. Everything that we associate with active, purposeful behavior in the animal kingdom, the ability to hunt, to navigate, to communicate, to think, traces back, ultimately, to the bilateral symmetry that first appeared in the Ediacaran. The organisms of the Ediacaran that showed bilateral symmetry, Dickinsonia, Kimberella, the early trace-making bilaterians, were not just interesting curiosities. They were the pioneers of a body plan that would eventually produce the entire diversity of bilateral animals, which includes the vast majority of animal species alive today. The third fundamental contribution of the Ediacaran was ecological complexity itself, the development of multi-species communities with structured ecological relationships, competitive and facilitative interactions, habitat engineering, and the other features of functioning ecosystems. Before the Ediacaran, the living world was, in ecological terms, relatively simple. Microbial communities had ecological structure. Certainly, they were not random assemblages, but the range of ecological interactions available to single-celled organisms is necessarily limited. With the appearance of multicellular organisms in the Ediacaran, ecological complexity took a quantum leap. Organisms of different sizes, different body plans, different ecological strategies began to interact with one another in ways that created genuinely complex ecological webs. Competition for space on the seafloor, facilitation between species, the modulation of local physical conditions by habitat engineering organisms, the early stages of predation, all of these ecological relationships were present in the Ediacaran. And the development of these ecological relationships was not just important for the Ediacaran itself. It was important for everything that followed because ecological complexity, once established, tends to be self-reinforcing. A diverse community creates more ecological niches than a simple one, because each species in a community potentially provides a niche for other species, as prey, as habitat, as competitor or mutualist. Diversity begets diversity. The ecological diversification of the Cambrian was not happening in an ecological vacuum. It was happening in a world that already had ecological structure, already had ecological relationships, already had the basic dynamics of multi-species community functioning. The Ediacaran had established the ecological framework. The Cambrian filled it in with an extraordinary proliferation of new forms and strategies. Let us now turn to one of the most profound and philosophically challenging aspects of the Ediacaran, its relationship to the concept of deep time. Because the Ediacaran forces us to confront deep time in a way that few other topics in science do, and confronting deep time changes something fundamental about how we understand our place in the history of life. Deep time, geological time, the 4 billion year span of Earth's biological history, is genuinely difficult to comprehend. Our minds evolved to deal with time on the scale of human experience: minutes, hours, days, years, perhaps centuries. The timescales of geology and evolution, millions and hundreds of millions of years, are so far outside our intuitive grasp that they become effectively meaningless. We say 538 million years, and the number slides off our consciousness without really landing. But the Ediacaran offers us a way to approach deep time that is more than merely numerical. It offers us a world, a real, detailed, ecologically functioning world that we can imaginatively inhabit. It gives deep time a face, or rather many faces: the fractal fronds of the rangiomorphs, the bilateral modules of Dickinsonia, the rasping traces of Kimberella. It makes deep time specific, textured, particular, and in doing so, it changes our relationship to the present. Because if we can imaginatively inhabit the Ediacaran, if we can make real to ourselves the fact that living, ecologically organized communities existed on this planet more than 500 million years ago, then we begin to see ourselves differently. We begin to see ourselves not as the culmination of life's history, not as the endpoint of a progressive march toward greater complexity and intelligence, but as one moment in a vastly longer story. A story that was already extraordinarily old and extraordinarily rich long before anything like us appeared. This is humbling in the best sense. Not humbling in the sense of making us feel small or insignificant. We are, after all, the products of 4 billion years of evolution, which is an extraordinary heritage, but humbling in the sense of making us aware of the vastness of the context in which we exist. We are not the first complex life. We are not the first ecological community. We are not the first experiment in multicellular organization or in behavioral complexity. We are the latest in a very long line of experiments, each building on the last, each dependent on all that came before. The Ediacaran organisms did not know that they were building the foundation for us. They did not have the foresight to see that their experiments in multicellularity, in bilateral symmetry, in ecological organization, would eventually produce, through hundreds of millions of years of subsequent evolution, beings capable of asking questions about the universe they inhabit. They simply lived, absorbing nutrients, growing, reproducing, dying in the world they found themselves in. And in doing so, they made us possible. That is a debt we cannot repay, but we can acknowledge it. We can look back across the vast distance of geological time and recognize, in the strange fractal fronds of the rangiomorphs and the quiet bilateral symmetry of Dickinsonia, something that we can call, without exaggeration, our beginning. Let us turn now to the specific fossil sites of the late Ediacaran, the sites that preserve the final chapter of the Ediacaran world, and try to read in them the story of the transition that was happening. The Nama group in Namibia is, as we mentioned in Part 1, one of the most important late Ediacaran localities. The rocks of the Nama group were deposited in shallow marine environments on what was then a continental shelf, and they preserve a diverse assemblage of organisms from approximately 545 to 538 million years ago, right at the boundary between the Ediacaran and the Cambrian. The Nama biota includes Cloudina with its mineralized shell and its bore holes. It includes Namacalathus, another mineralized organism, a goblet-shaped structure that sat on a stalk attached to the seafloor, with an open cup at the top that may have been the site of filter feeding. It includes Namapoikia, a larger organism that built a more complex mineralized structure, potentially similar to a sponge or a coral in its basic ecological role. These mineralized Nama organisms are ecologically different from the classic Ediacaran fauna of the earlier period. They are building structures. They are investing energy in hard parts. They are, in some cases, apparently filtering food from the water, a feeding strategy that is more active and more selective than the passive absorption of the rangiomorphs. They represent in many ways a transitional fauna, organisms that still have a distinctively Ediacaran quality in their overall simplicity and in the absence of the complex behavioral repertoire that characterizes Cambrian animals, but that are already moving toward the more energetically intensive, more actively defensive, more structurally complex biology of the Cambrian. The Nama group also preserves a different kind of trace fossil record from earlier Ediacaran sites. The traces at Nama are more diverse, more complex, and indicate more sophisticated burrowing behavior than the traces of earlier Ediacaran communities. By the time we reach the Nama biota, the transition is clearly well advanced. The mat world of the earlier Ediacaran has been significantly disrupted. The bioturbation that will eventually transform the seafloor completely is already well underway. And significantly, the classic Ediacaran organisms, the rangiomorphs, the vendobionts, organisms like the earliest forms of Dickinsonia, are absent or extremely rare at Nama. The older Ediacaran fauna had either disappeared or retreated to environments that are not preserved in the Nama record. The world had changed enough that the ecological conditions required by those organisms no longer existed in the environments where Nama was forming. The official end of the Ediacaran period is defined geologically by the beginning of the Cambrian period at approximately 538 million years ago. This boundary is defined primarily by the appearance in the fossil record of a trace fossil called Treptichnus pedum, a distinctive, complex burrowing trace that represents a level of behavioral and locomotor sophistication not seen in earlier Ediacaran trace fossil assemblages. Treptichnus pedum is itself a remarkable fossil. It is not the impression of a body, it is the trace of behavior, the record of an animal's movement through the sediment, preserved in the physical disruption of the sediment grains. The trace is complex. It shows a three-dimensional path through the sediment, with the burrowing animal apparently probing in multiple directions as it moved, searching for food. This is sophisticated burrowing behavior, burrowing with a purpose, driven by sensory information about where food is likely to be found. The appearance of Treptichnus pedum marks in the geological record the beginning of the Cambrian, but the boundary it marks is not a sharp one in biological terms. The transition from Ediacaran to Cambrian communities was not instantaneous. It was a process that played out over millions of years, with the gradual replacement of Ediacaran ecological modes by Cambrian ones. Treptichnus pedum is merely the paleontological marker for a transformation that was already well underway when the first T. pedum was made. Similarly, not all Ediacaran organisms disappeared at the Cambrian boundary. Some groups, particularly some of the mineralized organisms, seem to have survived into the early Cambrian at least for a time. And some characteristics of Ediacaran ecology, the passive filter feeding strategies, the preference for low-energy environments, the construction of simple colonial structures, persist in modified form in some Cambrian and later animal groups. The Ediacaran did not simply stop. It faded, transformed, was absorbed into the emerging world of the Cambrian. There is a concept in ecology called legacy effects, the way that the activities of one community of organisms can influence the conditions experienced by subsequent communities in the same place, even after the original community has been replaced. The Ediacaran world had enormous legacy effects on the Cambrian world that succeeded it. The most obvious legacy effect was the transformed seafloor. By the beginning of the Cambrian, the seafloor had been significantly altered by the burrowing activities of late Ediacaran organisms. The stable, layered structure of the microbial mat was disrupted. The chemical gradients that had characterized the Ediacaran seafloor had been altered. The physical structure of the sediment had been mixed and homogenized in ways that would persist and intensify as Cambrian burrowers became more diverse and more effective. This transformed seafloor was the substrate on which the Cambrian diversification took place. The Cambrian animals evolved in a physical environment that had been created, in part, by the biological activities of their Ediacaran predecessors. They inherited a world that had already been changed by life, and they continued to change it in the direction that those early changes had set. A second legacy effect was the evolutionary innovations that had been developed in the Ediacaran, and that formed the toolkit available to Cambrian evolution. The multicellular body plans, the bilateral symmetry, the developmental programs, the beginnings of tissue differentiation and organ systems. All of these were Ediacaran developments, biological innovations that arose in the context of the Ediacaran world and that were available for Cambrian evolution to elaborate and extend. Without the Ediacaran, the Cambrian explosion as we know it could not have happened. Or rather, something might have happened eventually. Life would presumably have continued to evolve, but it would have been a different kind of explosion, starting from a different biological foundation, producing different results. The specific character of the Cambrian explosion, the specific body plans that diversified, the specific ecological strategies that proliferated, was shaped by the specific biological toolkit that the Ediacaran had assembled. A third legacy effect was ecological. The Ediacaran had established the basic structure of marine ecological communities, the existence of producers and consumers, of competing and facilitative interactions, of the trophic relationships that organize energy flow through an ecosystem. The Cambrian diversification did not build this ecological structure from scratch. It elaborated and extended a structure that already existed, adding new trophic levels, new ecological roles, new kinds of interactions to a framework that the Ediacaran had established. The question of what happened to the Ediacaran organisms, where they went, whether they had any descendants, whether any of them managed to make it through the Cambrian boundary and into the later Paleozoic, is one that paleontologists have wrestled with for decades, and it still does not have a completely satisfying answer. For some Ediacaran organisms, the answer seems to be straightforward extinction without clear descendants. The rangiomorphs, with their unique fractal body plan and their passive osmotic absorption feeding strategy, have no clear descendants in the Cambrian or later fossil record. No Cambrian organism looks like a rangiomorph. No living organism has a body plan remotely similar to that of Charnia or Fractifusus. If the rangiomorphs left any descendants, those descendants had transformed themselves so completely that the relationship is no longer visible in the fossil record. For other Ediacaran organisms, the picture is murkier. Kimberella's possible relationship to the mollusks, if confirmed, would mean that the mollusk lineage traces back to the Ediacaran, a lineage that is, of course, extraordinarily successful in the modern world, including snails, clams, octopuses, and squid. If Kimberella is a primitive mollusk or a close relative of the mollusk stem group, then the Ediacaran has living descendants among the most familiar and diverse of modern marine animals. Similarly, the early bilaterians whose burrowing traces are preserved in the later Ediacaran record were presumably the ancestors of many Cambrian animal groups. The connection between those trace makers and specific Cambrian animals is not clear from the fossil record, but in principle, many Cambrian animal lineages, annelids, early arthropods, early echinoderms, should trace their ancestry back through the late Ediacaran. And there is the tantalizing question of the Cambrian small shelly fauna, the diverse assemblage of tiny, mineralized fragments that appears in the rock record just before and after the Cambrian boundary. These fragments, the microscopic shells and sclerites and spicules that appear in abundance in the earliest Cambrian rocks, represent the mineralized parts of organisms that were not yet preserving as complete body fossils. Some of these tiny mineralized fragments may represent the earliest Cambrian descendants of Ediacaran lineages, organisms that had taken the Ediacaran experiment in hard parts and carried it forward into the New World. The small shelly fauna is one of the most important and least understood assemblages in the fossil record. It is the biological bridge between the Ediacaran and the fully Cambrian world, the record of organisms that were evolving rapidly, experimenting with mineralization in dozens of different ways, producing forms that are often impossible to classify because they are so transitional, so in between. They are the products of evolution working at maximum intensity, under maximum selective pressure, producing maximum diversity in minimum time. And they are the proof, if proof were needed, that the Ediacaran did not simply end. It transformed. Its genetic toolkit, its ecological inheritance, its evolutionary momentum, all of these were carried forward in modified and elaborated form into the Cambrian. The Ediacaran organisms may have disappeared from the fossil record, but the evolutionary processes and innovations of the Ediacaran did not disappear. They were incorporated, transformed, and extended into everything that came after. let us take a long view now. Let us zoom out from the Ediacaran Cambrian transition and look at the broader sweep of life's history, and ask what the Ediacaran's place in that history tells us about the nature of major evolutionary transitions. The history of life is punctuated by what paleontologists call major evolutionary transitions, moments when the fundamental organization of life changed dramatically, when new levels of biological complexity were achieved, when the basic terms of ecological existence were transformed. The origin of life itself, the evolution of the eukaryotic cell, the evolution of multicellularity, the evolution of bilateral symmetry and complex developmental programs. The evolution of mineralized skeletons, the colonization of land, the evolution of endothermy, the evolution of social behavior and language. Each of these transitions changed the world profoundly and irreversibly. Each one opened up new evolutionary possibilities that had not existed before, and each one created a world in which some of what had existed before could no longer continue, in which the conditions that had sustained previous life forms were transformed by the innovation itself. This is the paradox of evolutionary progress. Every major advance in biological complexity tends to destroy the world that preceded it, replacing it with something more complex but also more competitive, more energetically demanding, less gentle in its ecological dynamics. The oxygenation of the atmosphere destroyed the world of the anaerobic microbes. The evolution of predation destroyed the world of the Ediacaran. The evolution of vision, hard parts, and sophisticated locomotion created a world of intense biological interaction that destroyed the more relaxed ecological dynamics of the late Ediacaran. And yet each of these destructions was also a creation. The oxygenated atmosphere created the conditions for complex eukaryotic metabolism. The end of the Ediacaran created the conditions for the Cambrian explosion. Every ending is also a beginning, and the story of life is the story of this continuous creative destruction, of worlds that arise, flourish, and then give way to the worlds that their own success created. The Ediacaran is the most ancient and the most complete example of this process that we have clear evidence for. It is the oldest chapter in the story of complex life, and it exhibits in its origin, its flourishing, and its ending the same fundamental pattern that runs through every major chapter in the history of life. It arose from the transformation of an earlier world, the cryogenian, the Snowball Earth events, the changed ocean chemistry that followed. It flourished under specific conditions that it helped to create and maintain. And it ended as the evolutionary process it had set in motion produced innovations, burrowing, predation, biomineralization, that transformed the conditions of its own existence. It is a complete story, not a tragedy, not a failure, but a complete and coherent chapter in the 4-billion-year narrative of life on Earth. Modern research on the Ediacaran is more active and more sophisticated than at any previous point in the history of paleontology. New fossil sites are being discovered regularly. New analytical techniques are being applied to known specimens. New theoretical frameworks are being developed for understanding the biology and ecology of these extraordinary organisms. Some of the most exciting recent research has focused on the molecular biology of the genes that controlled Ediacaran development. Even though we cannot observe living Ediacaran organisms, we can study their genetic legacy in the genomes of modern animals, particularly in the deeply conserved regulatory genes that are shared across the entire animal kingdom. By comparing the genomes of modern animals and reconstructing, through comparative analysis, what the ancestral genome of the earliest animals must have looked like, scientists are building an increasingly detailed picture of the genetic toolkit that was available in the Ediacaran. This work has revealed that many of the genes that control the development of complex animal features, the development of body axes, of cell type specialization, of organ systems, are extremely ancient. They predate the Cambrian explosion. They were already present in at least rudimentary form in the ancestors of all modern animals, ancestors that lived in the Ediacaran. This suggests that the developmental sophistication of Cambrian animals was not achieved by the invention of entirely new genetic programs, but by the elaboration and redeployment of programs that were already in place in the Ediacaran. This is one of the most profound insights to come from molecular biology applied to the deep evolutionary history of animals. It tells us that the Ediacaran was not just the ecological precursor of the Cambrian, it was the genetic precursor. The genome of every living animal carries, written in its regulatory DNA, the traces of the Ediacaran world. Every time a human embryo develops from a single cell into a complex bilateral organism with specialized tissues and organs, it is executing, in vastly elaborated form, programs that were first assembled more than 500 million years ago in the bodies of the first complex multicellular animals. You are, in this very literal sense, an Ediacaran organism's descendant. Your body, your bilateral symmetry, your complex organ systems, your nervous system, your capacity for experience and thought, all of these are the products of evolutionary processes that began in the Ediacaran. You carry the Ediacaran in your DNA. There is another dimension of the Ediacaran's relevance to the modern world that is worth exploring, and that is its relevance to our understanding of how ecosystems respond to major environmental changes. This is not a topic about current events, it is a topic about the deep patterns of life's history, but it is a topic with clear intellectual significance for how we think about the dynamics of complex living systems. The Ediacaran ecological transition, the gradual replacement of the mat-dominated, low bioturbation seafloor community by the bioturbated, predation-structured community of the Cambrian, is one of the best documented examples in the geological record of what ecologists call a regime shift. A regime shift is a relatively abrupt change from one stable state of an ecosystem to a different stable state, driven by the accumulation of changes that eventually push the system past a tipping point from which return to the previous state is very difficult or impossible. The Ediacaran to Cambrian transition appears to have had this character. For tens of millions of years, the Ediacaran ecosystem maintained itself in a stable state, the mat world, with its characteristic organisms and ecological relationships. Then, as bioturbation increased and predation intensified, the system began to shift. The shift was not linear. There were probably periods of relative stability and periods of more rapid change, but the overall direction was irreversible. The mat world did not return once it had been sufficiently disrupted. The new ecological state, the bioturbated seafloor, the predation-structured community, was stable in its own right and self-reinforcing. Understanding regime shifts in ecosystems is one of the most important challenges in modern ecology. Recognizing the warning signs of an approaching shift, understanding the feedbacks that drive and maintain different ecosystem states, developing ways to manage ecosystems so as to avoid unwanted transitions. These are active areas of ecological research with significant practical implications. The Ediacaran Cambrian transition is, from this perspective, a natural experiment of enormous scale and duration, a case study in ecosystem regime shifting that provides a deep time context for understanding processes that continue to operate in the living world today. Let us return, in these final sections of our documentary, to the places. To the physical reality of the Ediacaran fossil record, the cliffs and valleys and desert outcrops where the evidence of this ancient world is preserved, because science is not only done in laboratories and in the mind, it is done in places, and the places where Ediacaran fossils are found have their own character, their own beauty, their own power to connect us to the deep past. Mistaken Point on Newfoundland's windswept coast, where the fog rolls in from the North Atlantic and the ancient seafloor surfaces are exposed in the dark, fine-grained rock of the cliffs. Scientists worked there on their knees with hand lenses and notebooks, tracing the outlines of organisms that lived before there were eyes anywhere on Earth, in a landscape of extraordinary austere beauty. The connection between the physical reality of the place, the cold, the fog, the ancient rock, and the creatures preserved within it is something that no description can fully capture. You have to stand there. You have to put your hand on the rock and feel the surface where the rangeomorphs grew, and try to hold in your mind simultaneously the present reality of the exposed cliff and the ancient reality of the seafloor 565 million years ago. The Flinders Ranges in South Australia have their own quality, a dry, sun-baked landscape of folded and eroded ancient rock, in colors ranging from red to ochre to pale gray, where the Ediacara Hills rise from the desert plain. The fossils here are found in cream-colored sandstones and siltstones, and the organisms themselves are often preserved as convex structures on the undersides of rock layers, casts of organisms that were pressed into the seafloor sediment and then buried by successive layers of sand. To hold an Australian Ediacaran fossil specimen, a Dickinsonia or a Triberchidium preserved on the undersurface of a sandstone slab, is to hold something that was alive when life on Earth was young in a way that we can barely comprehend. The White Sea coast of Russia, where the Ediacaran fossils are found in ancient sedimentary sequences exposed by the rivers draining into the sea, has a different quality again, a vast northern landscape of boreal forest and tundra, where the fossils emerge from the river banks in summer, when ice and snow have retreated and the ancient rock is briefly accessible. Russian paleontologists working on the White Sea material have contributed enormously to our understanding of the Ediacaran biota, describing dozens of new species and developing many of the interpretive frameworks that we use today to understand these organisms. Each of these places is different. Each preserves a somewhat different window into the Ediacaran world. And together they give us something that no single site could provide, a sense of the geographical scope and the biological diversity of the Ediacaran world. The fact that this was not a local phenomenon, not a regional curiosity, but a genuinely global episode in the history of life, playing out simultaneously in what are now 5 different continents, across hundreds of millions of years, in a world that was geographically very different from the one we know. The continents in the Ediacaran were not in their current positions. Plate tectonics, the slow movement of the continents across the surface of the Earth, driven by convection in the underlying mantle, has been operating continuously for billions of years, and the configuration of the continents in the Ediacaran was radically different from today. Most of the major continents of the Ediacaran world were part of a supercontinent called Rodinia, which had been assembled prior to the Ediacaran and was in the process of breaking up during the period. The breakup of Rodinia created new shallow marine environments, the continental shelves that form as continents separate and ocean basins open between them, and these new shallow environments were the ecological stage on which the Ediacaran drama played out. The creation of extensive shallow water environments with their relatively warm, well-lit, oxygenated water may itself have been one of the triggers for the Ediacaran diversification, providing the ecological space into which the new multicellular organisms could expand. As the Ediacaran progressed, the continents continued to move. The ocean basins changed their shapes. The circulation patterns of the ocean changed, affecting the distribution of nutrients and oxygen. The climate shifted. There were significant climatic changes within the Ediacaran, though nothing as extreme as the Snowball Earth events that preceded it. All of these physical changes created a dynamic backdrop against which biological evolution was playing out. Understanding the tectonic and climatic context of the Ediacaran is important for building a complete picture of the period. The organisms we have been discussing did not live in a physical vacuum. They lived in a specific physical environment, and that environment was changing on timescales that overlapped with the timescales of biological evolution. The relationships between tectonic change, climatic change, oceanographic change, and biological evolution in the Ediacaran are complex and not fully understood, but they are an important frontier of current research. We are approaching the end of our exploration of the Ediacaran, but before we finish, let us spend some time with the question that is, perhaps, the most important one of all. What does the Ediacaran mean? Not in a scientific sense, we have been discussing the scientific meaning throughout, but in a deeper sense. What does it mean that life worked this way? What does it mean that there was a world before eyes, before teeth, before the arms race of predator and prey? What does it mean that this world existed, flourished, and then gave way, not suddenly, not catastrophically, but gradually, systematically, through the internal logic of its own biological success? The Ediacaran means that life's history is far richer and stranger than we tend to imagine. We have a tendency to think of life's history as a linear progression, from simple to complex, from primitive to advanced, from microbial to animal to intelligent. The Ediacaran disrupts this linear story. It shows us that the road to complexity was not a single straight road, but a branching network of paths, most of which led to evolutionary dead ends, and some of which connected to the mainstream of subsequent evolution in ways that are not always obvious. The rangeomorphs tried one path and found that it did not lead anywhere further. Dickinsonia tried another path and may have contributed something to the animal lineage, even if it left no clear descendants. Kimberella tried a path that led perhaps to the mollusks. The early bilaterians tried paths that led to the Cambrian explosion. All of these paths were genuine experiments. All of them were viable in their own contexts. Not all of them were viable in the context that followed. This is the nature of evolution. It is not a goal-directed process. It does not know where it is going. It simply follows the gradient of reproductive success in the current environment, producing innovations that work now, with no capacity to predict whether those innovations will work in future environments. The rangeomorphs were not failures. They were successful in their time and place. The fact that their time and place eventually gave way to different conditions is not a reflection on them. It is a reflection on the nature of time itself, on the fact that nothing in the physical world is permanent, that all conditions are temporary, that even the most successful biological form is ultimately contingent on the maintenance of the conditions that made it possible. This is a deeply important insight, and the Ediacaran brings it home in a way that more familiar examples of extinction cannot quite match. The dinosaurs went extinct, we tend to think, because of a catastrophic event, an asteroid, a series of volcanic eruptions, a collapse of the ecosystem they depended on. The Ediacaran organisms did not go extinct in that sense. They were not wiped out by an external catastrophe. They were outcompeted gradually by the consequences of their own world's biological activity. Their extinction was, in a sense, biological rather than physical. It was the natural consequence of a living world that kept evolving, kept innovating, kept creating new pressures and new opportunities. A world that could not stand still, even if standing still might have been, for the organisms of the Ediacaran, the optimal strategy. The Ediacaran also means something about our relationship to ancient life, about what we owe, intellectually and imaginatively, to organisms that existed before we did, and that made us possible. We are accustomed to thinking of our relationship to the past in terms of our own species, in terms of human history, human ancestors, the long chain of cultures and civilizations that preceded us and shaped us. But the Ediacaran pushes that relationship of precedence and foundation back much further, back before human beings, before mammals, before vertebrates, before fish, before the Cambrian explosion, to the very beginnings of the kind of complexity that eventually produced us. The rangiomorphs are not our ancestors in any direct biological sense. They appear to have left no descendants in the modern world, but they are our predecessors in the most fundamental sense. They existed first. They occupied the world of complex multicellular life first. They worked out through billions of years of evolutionary history the fundamental problems of being a large, complex multicellular organism, the problems of coordination, of feeding, of reproduction, of ecological interaction. They did not solve all those problems in the ways that we inherited, but they tackled them first. And Dickinsonia and Kimberella and the early bilaterians whose burrowing traces are preserved in the ancient sediment, these are our ancestors in a more direct sense. They are on the line of descent, or close to it, that eventually led to us. They carry in their body plans and their biological organization the seeds of the bilateral symmetry, the tissue complexity, the directional behavior that evolution would eventually elaborate into the extraordinary biological machinery of the vertebrate body. To look at a Dickinsonia fossil and try to imagine it alive is to look at a very distant ancestor. Distant in time more than 500 million years, and distant in biological complexity, but ancestor nonetheless. The same evolutionary process that shaped Dickinsonia eventually shaped you. The same fundamental biology, DNA, proteins, cells, the biochemical machinery of life operated in both. You are made of the same stuff that Dickinsonia was made of, organized by processes that trace their origins to the same ancient chemistry. The final lesson of the Ediacaran, perhaps the deepest lesson, is about what we value in the history of life and why. We tend to value success. We tend to celebrate the organisms and the lineages that survived, that diversified, that eventually gave rise to the remarkable complexity of the modern living world. We celebrate the Cambrian as the explosion that produced everything. We celebrate vertebrates for their evolutionary success. We celebrate implicitly the lineages that led to us because we are the ones doing the celebrating. But the Ediacaran asks us to broaden our sense of what counts as worth valuing in life's history. The rangiomorphs did not lead to us. They did not lead to anything that is alive today, as far as we know. By the metric of reproductive success across geological time, they were failures. But by any other metric, by the metric of ecological innovation, of biological novelty, of genuine creativity in the face of the deep evolutionary problem of how to be a large, complex organism, they were extraordinary successes. They solved in their own way the problem of building a large, complex body in an ancient ocean. They solved it in a way that worked beautifully for tens of millions of years, in a specific ecological context that they helped to create and maintain. They filled the Ediacaran world with their fractal beauty, their ecological complexity, their patient chemical existence. And then when the world changed, they could not follow it. Is that a failure? Or is it simply what it means to be a living thing in a changing world, to be adapted to the present, to flourish in the present, and to be unable to follow every future direction that the present might unfold into? We should not pity the rangiomorphs for their extinction. We should not regard the Ediacaran as a failed experiment or as a world that didn't quite make it. We should regard it as what it was, a complete world, a world that was real and functioning and ecologically sophisticated in its own right, a world that existed for nearly 100 million years, longer than the entire history of the primates, and that, in existing, in flourishing, and eventually transforming, made the world we inhabit possible. There is a particular quality to the experience of thinking about the Ediacaran that is different from the experience of thinking about most other aspects of ancient life. When we think about the dinosaurs, we feel excitement. They were large, they were dramatic, they were in some ways superficially familiar, because they moved and hunted and competed in ways that we can imaginatively inhabit. When we think about the Cambrian, we feel wonder at the diversity of forms, the extraordinary morphological experimentation, the bizarre anatomy of organisms like Anomalocaris and Hallucigenia and Opabinia with its 5 eyes and its flexible feeding tube. When we think about the Ediacaran, the experience is different. It is quieter. It is slower. It has a quality of deep stillness, of patience, of things happening on timescales that make human attention difficult to maintain. The rangiomorphs did not run or hunt or hide or sing. They grew, they absorbed, they swayed in the current. They were beautiful in the way that a mathematical structure can be beautiful, precise, elegant, self-consistent. And that quiet beauty, that slow, chemical, patient existence has something to teach us about what it means to be alive. Not everything that is living has to be dramatic. Not everything that matters has to be fast or loud or competitive. The Ediacaran was alive in ways that are completely unlike our experience of being alive, and yet it was genuinely, fully alive, participating in the great biological experiment of existence, contributing to the ongoing evolutionary process, leaving its mark on the world in ways that persist to the present day. Every time you look at a living thing, any living thing, no matter how simple or complex, you are looking at something that carries within it the traces of a history stretching back 4 billion years. Back through the Cambrian and through the Ediacaran, back through the Cryogenian and through the long billions of years of microbial existence, back to the very origin of the first self-replicating molecules in the ancient chemistry of the early Earth. Every living thing is a library of that history, written in molecular language, carrying forward the lessons and innovations and accidents of an unimaginably long journey. The Ediacaran is a chapter in that library, a long chapter, a beautiful chapter, a chapter full of organisms that we have only recently begun to read with any comprehension. And the more carefully we read it, the more fossils we find, the more analytical tools we apply, the more sophisticated our interpretive frameworks become, the richer and stranger and more wonderful it reveals itself to be. As we come to the end of this documentary, let us do what the best explorations of deep time always eventually do. Let us come back to the present. Let us stand in the present moment, wherever you are watching this, and let the Ediacaran reverberate through your understanding of where you are and who you are. You are watching this on a planet that is 4.5 billion years old. You are a biological entity whose organizational complexity traces back, through an unbroken chain of evolution, to the first complex multicellular organisms of the Ediacaran. The cells of your body are the descendants of cells that once made up organisms like Dickinsonia. The bilateral symmetry of your body, the left-right mirror image, the front-back orientation, the concentration of sensory organs at the leading end, is an evolutionary inheritance from the first bilaterally symmetrical animals of the Ediacaran. The capacity you have to ask questions about the world, to wonder, to inquire, to build scientific understanding of the deep past, is the product of a brain that is the end result of the same evolutionary process that built the first nervous systems in the late Ediacaran and early Cambrian. The consciousness with which you are experiencing this moment, the subjective quality of your experience right now, as you listen to these words, may trace its ultimate origins to the first biochemical processes in early animals that constituted the biological beginning of experience. You are, in this very concrete and non-metaphorical sense, the Ediacaran's legacy. You are what happened when the experiments of the Ediacaran were continued, elaborated, and extended through 538 million more years of evolution. You are the latest expression of a creative process that began when the first multicellular organism first coordinated its cells into something more than a mere collection of individual units. That is not a small thing. That is, if we allow ourselves to feel the full weight of it, one of the most extraordinary facts about existence that we can contemplate. We are ancient. We are, in our biology and in our evolutionary history, far older than we tend to imagine. Our roots go down through geological time into a world that was already complex and ecologically sophisticated before the first shell had ever been built, before the first eye had ever opened, before the first tooth had ever been bared in the first act of predation. The Ediacaran period ended approximately 538 million years ago. But in another sense, in the sense that matters most, the sense of evolutionary inheritance and biological continuity, it has never ended. It lives on in the genomes of every animal on Earth. It lives on in the basic organizational principles of every multicellular body. It lives on in the ecological relationships of marine communities that have been shaped, however indirectly, by the transformations that began in the Ediacaran. And it lives on in our capacity to know it, in the scientific enterprise that has spent the better part of a century working to recover and understand the evidence of this ancient world, reading the rocks of Newfoundland and South Australia and Russia and Namibia and China, applying new analytical tools to old specimens, developing new theoretical frameworks for understanding organisms and ecosystems that have no living equivalents. The story of the Ediacaran is a story about life's capacity for genuine novelty, for trying things that have never been tried before, for building forms and strategies and ecological relationships that did not exist before and that may not exist afterward. It is a story about the relationship between life and the physical world that contains it, about the way organisms shape their environment and are shaped by it, in a reciprocal dance that has been going on for 4 billion years. It is a story about endings that are also beginnings, about the creative destruction that seems to be an inherent feature of life's history on this planet. And it is a story about us, about our origins, our evolutionary inheritance, our biological nature. The rangiomorphs are our predecessors. Dickinsonia is our ancestor, or something very close to it. The Ediacaran world is the world in which the biological toolkit that eventually produced us was first assembled. We came from this. This quiet, patient, fractal chemical world, this world of slow movement and passive absorption and ecological complexity built on microbial mats. This world before eyes, before shells, before the great noisy violence of predation that would eventually produce the modern ocean. We came from this, and knowing that, really knowing it, feeling it as more than an abstract fact, changes something in how we experience being alive. It deepens the experience. It connects it to something vast and ancient and strange. It makes the ordinary miracle of a living body, your living body, sitting or standing wherever you are, the heart beating, the lungs breathing, the neurons firing, feel like what it actually is. The latest expression of a 4 billion-year-old process of becoming. The Ediacaran is over. The world it made is gone. But what it built in the genomes and body plans and ecological relationships of the living world persists. Everything that came after the Ediacaran is, in the deepest sense, the Ediacaran's continuation. And we, watching this documentary, asking these questions, reaching back across 500 million years of geological time to try to understand what was there, we are the most recent and perhaps the most surprising expression of what the Ediacaran started. The first complex life looked at the world in the only way available to it, through chemistry, through contact, through the slow, patient process of growth and absorption. It could not know what it was starting. It could not know that what it was doing would eventually produce beings capable of looking back and knowing it, but it was starting it nonetheless. Every fractal frond of every rangiomorph, every bilateral module of every Dickinsonia, every rasping mark of every Kimberella, every tiny nested cone of every Cloudina, all of it was the beginning of something that would eventually become capable of understanding its own beginning. That is the Ediacaran, that is its meaning. That is why it matters and why it will always matter as long as there are minds capable of asking where they came from and what came before them. This has been the story of the world before everything we know, the forgotten world of Earth's first complex life. Thank you for watching. Please let us know in the comments where you are watching this history video from and what time you are watching it. Every single comment connects us across distance and time and reminds us that curiosity about the deep past is something that people everywhere share. And please subscribe to the channel if you haven't already. There are many more stories from Earth's extraordinary history waiting to be told, and we want to tell them with you.